There is no warning in the room when the danger first appears. The mother may feel perfectly well, her child still moving beneath her ribs with the quiet insistence of life. Nothing in the shape of her body suggests that something has crossed an invisible boundary, yet in a narrow tube of blood, in a laboratory far from the intimacy of the womb, a sign emerges: antibodies, made by the mother herself, capable of finding the red blood cells of her unborn child and marking them for destruction. The threat does not arrive from poison, fever, hunger, or injury. It comes from a difference so small that for almost all of human history no one could see it, name it, or understand why one pregnancy ended in joy, while another in the same family ended in bewildering loss. A woman can carry a child whose blood bears a surface marker her own blood does not. Her immune system, built to protect her, can encounter that difference and remember it, later, in another pregnancy. That memory can become a weapon. Long before the language of antigens and antibodies existed, this hidden encounter must have unfolded in homes, huts, farms, palaces, and crowded city rooms across generations, leaving only grief and a question with no vocabulary yet to hold it. Today the difference is commonly reduced to a symbol on a medical chart, a plus sign or a minus sign beside a blood group. O positive, a negative, AB negative. It looks almost administrative, the kind of detail printed on a hospital bracelet or entered into a form and forgotten until needed. But the minus sign can carry a strange gravity. It refers, most often in everyday speech, to the absence of the D antigen within the Rh blood group system. A feature on the surface of red blood cells that many people possess and some do not. To be Rh negative is not to be ill. It does not make a person fragile, alien, gifted, cursed, or separated from humanity by some hidden border. Most people who carry Rh negative blood live without ever feeling its absence. The body does not announce it. There is no unusual heartbeat, no distinctive eye colour, no secret sensation beneath the skin. The mystery begins precisely because this difference is silent until blood meets blood under particular conditions: a transfusion, an injury, a birth, a miscarriage, or a pregnancy in which mother and fetus do not share the same RHD status. Imagine that story before the age of modern testing. A first child is born alive and apparently healthy. The family takes this as a promise of more children to come. Then a later infant is born desperately pale or yellowed, weak in the arms, swelling with fluid before birth, or failing in the days after delivery. Perhaps there are several losses. Perhaps the mother is blamed, or fate is blamed, or God, or poor air, or bad luck, or an inherited weakness no one can identify. In many cases, even the pattern would remain obscured, because pregnancy loss is common for many reasons, and the past preserves its private tragedies unevenly. No archive records every baby who never drew breath, every midwife who saw the same sorrow return, every family tree with its blank spaces. The evidence remains incomplete. Yet what survives in modern medicine allows a haunting inference for countless families. The cause was not visible disease, but an immune conflict created by inheritance. One that could only be understood once human beings learned that blood is not a single universal substance, but a crowded landscape of molecular identities. That is the real doorway into the R-negative story, and it is more unsettling than the legends that later gather around it. Blood has always carried symbolic weight. It means kinship and sacrifice, purity and contamination, royal inheritance and ancestral belonging. When science eventually identifies a rare difference in blood, especially one capable of affecting pregnancy, it enters a world already prepared to turn biology into mythology. The words negative and rare do not remain neutral for long. They begin to suggest a missing ingredient, an older lineage, an unusual origin. Where the trait occurs more frequently in certain populations, people begin to imagine isolated survivors of forgotten eras, where medicine speaks in technical language about incompatibility, antibodies, and fetal danger. Other voices hear a story of bloodlines that cannot peacefully mix. And where science proceeds with the frustrating caution of probabilities, incomplete population histories, and genes altered by ordinary evolution, mythology offers something cleaner: a secret, a chosen group, a hidden past, perhaps even an institution determined to conceal the truth. But before the internet forums, before the speculative documentaries, before claims of psychic sensitivity, ancient visitors, or powers supposedly carried in the veins, there is the human fact at the centre of the story. A mother and child can be biologically connected and immunologically mismatched. The fetus receives its genetic inheritance from two parents. If the mother lacks the RHD antigen and the child inherits it from the father, small amounts of fetal blood may sometimes enter the maternal circulation, especially around childbirth, but also during certain events in pregnancy. The mother's immune system may then recognise those fetal red cells as unfamiliar and produce antibodies against the D antigen. That first encounter does not always devastate the child who triggered it. The most severe danger often comes later, after the immune system has learned what to look for. In a subsequent pregnancy with another RHD-positive fetus, maternal antibodies can cross the placenta and attack fetal red blood cells. The child's own bloodstream becomes the site of a quiet, escalating struggle cells, destroyed faster than they can be replaced. Anemia deepening, organs straining to compensate, the body retaining fluid in the worst cases. This condition, now known as hemolytic disease of the fetus and newborn, is one of those medical realities whose calm modern name barely contains the fear it once carried. It is also a story transformed so dramatically by prevention that many people in wealthy medical systems no longer encounter its full severity. An injection of anti-D immune globulin, offered to eligible Rh-D-negative pregnant patients at carefully chosen times, can prevent the maternal immune system from becoming sensitized in the first place. Yet its very success hides the magnitude of what came before. Until the 20th century, there was no protective injection. No routine screening early in pregnancy, no way to monitor fetal anemia with modern imaging, no safe framework for explaining why an apparently healthy mother repeatedly lost Rh-positive infants. Even now, access to prevention and specialist care is not equal everywhere, and other blood group antibodies can produce similar danger. The mystery has not vanished. Much of its most tragic expression has simply been pushed back by knowledge. Before we go deeper, take a moment to like the video and subscribe, if this is the kind of history and mystery you enjoy. Because this is not ultimately a story about supernatural blood, it is a story about the enormous distance between an invisible biological event and the meanings human beings attach to it. For most of history, the first distance was impossible to cross. People could observe birth, illness, jaundice, weakness, and death, but they could not watch antibodies form. They knew families carried certain misfortunes and that blood mattered when it spilled from the body. They did not know that a red cell could bear microscopic markers inherited as surely as the shape of a nose, or that a mother's immune memory might be awakened by traces of her child's blood. In that darkness, repeated loss had no mechanism, and without mechanism there was room for fear. Modern mythology takes that old darkness and rearranges it, claiming that the lack of an antigen is evidence of a concealed ancient origin. The older truth is sadder and more powerful. A trait can be entirely natural and still shape the survival of children for millennia. The name itself contributes to the confusion. Negative sounds, as though something essential has been taken away. It invites the image of a bloodline defined by absence, an unfinished human type or a remnant from a separate branch of existence. But absence in genetics is not automatically deficiency. Human variation is full of features present in some people and absent in others: cell surface molecules, enzyme variants, immune system signatures, tolerances and susceptibilities distributed through populations by inheritance, chance, migration, isolation, and the relentless sorting of generations. A person with Rh-negative blood is not missing humanity. They are carrying one version of a normal human variation. What makes this variation compelling is not that it lies outside the human story, but that it reaches directly into some of the deepest forces within it: reproduction, ancestry, population movement, the hazards of childbirth, and the long struggle to turn pattern into explanation. Still, the emotional pull of the legends is easy to understand. Rarity can feel like identity. A blood type is intimate, inherited, and invisible. It seems to connect a person with ancestors they will never know. When a trait is unevenly distributed across the world and appears in striking proportions among populations already surrounded by stories of antiquity and isolation, it becomes tempting to read the map as a coded message. Perhaps, the speculation goes, our negative people descend from a lost civilization, from unusually ancient Europeans, from survivors of a catastrophe, from beings not fully human. Such theories promise that a clinical detail printed on a blood test is really a fragment of epic history. They also frequently borrow the suffering caused by Rh incompatibility, twisting a medical problem into an argument that two supposed bloodlines were never meant to combine. No credible genetic or medical evidence supports that conclusion. The child in danger is not proof of species separation. It is evidence of how an otherwise ordinary inherited difference can provoke the immune system when fetal and maternal blood cross paths. And yet dismissing the legends is not the same as dismissing the mystery. There are genuine questions here, questions more subtle than conspiracy and more enduring than fantasy. Why does the Rh-D negative phenotype occur at different frequencies in different populations? What prehistoric migrations and isolation shaped its distribution? Did it ever carry some advantage or cost beyond pregnancy incompatibility? Or did genetic drift do much of the work? How did a biological trait that can endanger later pregnancies persist at substantial frequencies in some parts of the world? Scholars still debate parts of that evolutionary history, because ancient populations do not leave complete genetic ledgers behind them. Bones decay, communities merge, migrations erase their own tracks, and modern frequencies are not simple photographs of the distant past. The uncertainty is real. It is simply not evidence of suppression. At the beginning, then, there is no secret laboratory and no hidden dynasty. There is a delivery room before anyone knows what to test for. There is a mother surviving a birth, perhaps relieved to hold one living infant, unaware that a few exchanged cells may have left a memory in her blood. There is a later cradle that remains painfully still, or a newborn whose skin turns yellow beneath anxious eyes, while everyone around the bed lacks the language to understand the enemy. The rare bloodline begins not as a tale of power, but as a mark that cannot be seen, a difference that is harmless in one life and devastating in one particular meeting of lives. Centuries will pass before medicine recognizes the pattern. Longer still before that pattern is surrounded by tales of ancient inheritance and concealed truth. But the first mystery is already complete, quiet and immense in the bond between mother and child. Blood can carry both belonging and danger, and the difference between them may be no larger than a molecule on the surface of a red cell. For the families who endured such losses, blood offered no explanation. It was visible only when it escaped the body bright on linen after childbirth, darkening on a surgeon's hands, pooling beneath the wounds of soldiers or hunters, drying on the floor of a room where life had just changed. Blood was understood as essential, certainly, but its hidden structure lay beyond every eye. A person could lose too much of it and die. A woman could bleed during labour and fade in a matter of hours. A newborn could arrive strangely yellow, weak, or swollen, and no one could connect that child's condition to a microscopic difference inherited from the father and encountered by the mother's immune system. Across much of human history, blood was less a biological system than a powerful symbol and a terrifying substance, warmth leaving the body, ancestry passing through generations, vitality that could be spilled but not replaced. The idea that one person's blood might be fundamentally incompatible with another's would have seemed both obvious in a symbolic sense and impossible in a medical one. Blood belonged to individuals and families in myth and lore, but physicians could not yet prove that it came in distinct types. Ancient healers did what all humans do when confronted with repeated suffering they observed, compared and built explanations from the tools they had. In many medical traditions, blood was tied to balance within the body. In Greek and later European medicine, it became one of the humours, associated with temperament and health, something that could accumulate in dangerous excess and be deliberately removed. Bloodletting survived for centuries not because physicians were indifferent to suffering, but because their theory of disease made the removal of blood appear rational. Fever, inflammation, confusion, pain, even difficult childbirth might seem to call for the opening of a vein. The bitter irony is that people already weakened by infection, injury, or delivery were sometimes deprived of the very fluid they most needed to survive, yet without microscopes capable of revealing cells. Without germ theory, without a working understanding of circulation or immunity, a body failing after loss of blood was interpreted within a world of balances, qualities, and corrupted fluids. The first great turn comes when blood ceases to be treated merely as something that collects or drains, and is instead understood as something moving continuously through the body. In the early 17th century, William Harvey demonstrates that the heart drives blood through a closed circulatory system. His insight changes medicine's map of the human interior. Blood is no longer a substance endlessly produced and consumed in different tissues. It moves, returns, circulates. That discovery creates a dangerous and irresistible possibility. If a dying person has lost blood, and blood travels through vessels, perhaps blood can be put back in. The idea appears almost simple once circulation is recognised. It is also the beginning of a long sequence of experiments in which hope repeatedly outruns knowledge. Within decades, investigators attempt transfusions between animals, and then between animals and humans. The scenes are difficult to imagine without the later cleanliness of a hospital ward. There are tubes made from quills, silver, or other improvised materials, restrained animals, physicians working without an understanding of sterile technique, patients already desperately ill, and blood transferred not by modern bags and measured infusions but through uncertain arrangements of vessels and pressure. In 17th century Europe, lamb's blood is infused into human recipients, partly because the animal is associated with gentleness and purity, as though character might travel in the veins along with blood. Some recipients survive small volumes, perhaps because little is actually transfused. Others develop fever, pain, dark urine, distress, collapse, or death. The results are confused by poor records, contamination, illness already present in the patient, and the complete absence of knowledge about immune reactions. Experiments become controversial and, in some places, are restricted or abandoned. What those early transfusion pioneers cannot know is that they are not merely moving nourishment or vitality from one creature into another. They are introducing foreign red blood cells covered in molecular markers that the recipient's body may attack with astonishing speed. The incompatibility between species makes the danger profound. But even transfusions between human beings will eventually reveal hazards no one yet suspects. Blood looks alike. One drop of human blood resembles another so completely to the naked eye that the idea of lethal internal differences seems almost perverse. Red is red, warm is warm, yet beneath that sameness lie signatures capable of turning attempted rescue into catastrophe. For generations, transfusion remains an uncertain practice, used rarely and often in extremity. The demand for it, however, never disappears, because hemorrhage never disappears. Childbirth remains one of the most urgent settings. A labour can proceed normally until within minutes, a mother begins losing blood faster than anyone can stop it. In the early 19th century, the English obstetrician James Blundell confronts women dying from postpartum hemorrhage and reasons that human blood, rather than animal blood, should be used to replace what has been lost. Beginning in the 1810s, he performs human-to-human transfusions, devising apparatuses to collect and deliver blood. Some patients survive, some do not. His work is careful for its time, and his conviction that only human blood should be given to humans is an important advance. But the results remain unpredictable. A transfusion that seems to revive one woman may bring no rescue to another, or may contribute to a rapid deterioration that no physician can explain. Here the Rh negative story moves through a shadowed corridor. A woman dying from hemorrhage might be saved by the blood of another person, but only if the match between them is sufficiently safe. A newborn destroyed by maternal antibodies might look, to physicians of the time, like an entirely separate tragedy. The two problems share an invisible language of blood cell recognition, but no one can yet read it, even when doctors begin describing patterns of jaundice and severe anemia in infants. They have no means of connecting those patterns to maternal sensitization. A baby may be born with a grave illness known through its outward appearance, while the cause remains hidden in the mother's blood. In older records, terms describing newborn jaundice, swelling, or weakness may encompass many different diseases. Some cases likely arise from Rh incompatibility, others do not. No single explanation fully accounts for every historical infant death. And it would be reckless to rewrite the past as though all unexplained losses came from one blood trait. Still, among the thousands of births recorded only as tragedy, Rh disease must sometimes have been present, working without a name. The 19th century makes blood increasingly measurable. Microscopes improve. Red cells can be examined as cells rather than imagined as a uniform fluid. Scientists study clotting, circulation, anemia, and the changes caused by disease. Surgeons and obstetricians become more interested in transfusion, especially as medical practice gradually grows more systematic. Yet transfusion remains frighteningly unreliable. Doctors know that fresh human blood can sometimes restore a patient after severe bleeding. They also know that it can produce violent reactions, chills, fever, pain, breathlessness, collapse, urine darkened by the destruction of blood cells. Some blame air entering the veins, some suspect clotting, weakness of the recipient, delay in transferring the blood, or flaws in the equipment. Each explanation may occasionally matter, but none captures the fundamental problem. The donor and recipient may both be healthy, both human, and still carry blood that cannot be safely combined. The crucial clue appears not through ancient lineage or dramatic archaeological discovery, but through an unglamorous laboratory observation. At the turn of the 20th century, the Austrian physician Karl Landsteiner studies what happens when blood from different people is mixed. He separates blood into its red cells and liquid serum, then combines samples from different individuals. Sometimes nothing alarming occurs, sometimes the red cells clump together visibly. A reaction called agglutination. The result is simple enough to observe, but profound in its implication. Human blood is not universally interchangeable. The serum of one healthy person may contain substances that react against the red cells of another healthy person. The danger of transfusion is not merely poor technique, it is built into biological variation. From those reactions emerges the classification eventually known as the ABO blood group system. Some people carry M markers on their red blood cells, some carry B, some carry both, and some carry neither. Their plasma contains predictable antibodies against markers they do not possess. This is not yet the Rh system, and the difference matters. An incompatible ABO transfusion can produce an immediate and often catastrophic reaction, because the relevant antibodies are commonly already present. Our incompatibility operates differently in many cases. A person who lacks the important D antigen generally must first be exposed before developing anti-D antibodies, which is why pregnancy and previous transfusion become so significant. But the discovery of ABO changes the entire intellectual landscape. It proves that red blood cells bear inherited identities invisible to ordinary sight. It turns transfusion from a desperate gamble toward a procedure that can be tested and planned. The effect is dramatic. When donors and recipients are matched for ABO type, transfusion becomes far safer, particularly as improvements in anticoagulants, storage, sterile practice, and wartime medical organisation allow blood to be collected and delivered with increasing reliability. The First World War, with its industrial scale of injury, accelerates the practical use of transfusion. Men arrive at aid stations pale from blood loss, their pulses barely detectable. And stored blood can now pull some of them back from the edge. Behind the apparent miracle lies a new respect for blood's hidden boundaries. A label attached to a bottle can determine whether it saves or kills. For the first time, the notion of blood compatibility begins to enter routine medicine rather than remaining a mystery observed only after disaster. And still, certain reactions refuse to disappear. A transfusion may be correctly matched according to the known ABO categories, yet the patient can develop unexpected trouble, especially after earlier transfusions. Likewise, doctors continue to see newborn infants with severe jaundice and anemia in patterns that ABO alone cannot adequately explain. Some infants are born swollen and desperately ill, some die before delivery, some appear healthy at birth and decline rapidly afterward. The disease acquires clinical description before it acquires its cause. Physicians can recognize that the infant's red cells are being destroyed, but the origin of that destruction remains elusive. The mother herself may appear healthy. Her pregnancy may seem normal until the child suffers. What survives in the medical record suggests that practitioners are circling a hidden mechanism, seeing its consequences from several directions without yet identifying the mark that triggers it. This is the strange interval in the history of blood knowledge has advanced far enough to expose a mystery, but not far enough to settle it. The old belief that all human blood is essentially alike has already broken apart. Doctors now understand that some incompatibilities can be predicted, and that inherited blood groups influence survival in the operating room and the maternity ward. Yet the newly discovered ABO system is not the whole answer. There are patients whose blood behaves as though another boundary remains hidden. There are pregnancies in which a mother's body seems to carry a lethal memory that cannot be explained by the categories already known. The evidence points toward an additional signal on red cells, another inherited distinction, silent in ordinary life and dangerous only when circumstances bring it into conflict. For the people living through those years, none of this uncertainty is abstract. It is a surgeon holding a bottle of donor blood and knowing that a careful match reduces danger but does not eliminate it. It is an obstetrician facing a mother whose first child lived and whose later infants have not, with no prevention to offer and only incomplete theories for comfort. It is a family told that a newborn has severe jaundice. While beneath the yellow skin, red blood cells are being destroyed in a battle no one can yet stop. The ancient symbolism of blood has not vanished, but it has been joined by something colder and more exacting, laboratory evidence that blood carries differences capable of deciding life and death. For centuries, people had looked upon blood and seen one substance, sacred or polluted, strong or depleted, spilled or inherited. Now the surface of that idea begins to fracture. A drop of blood can belong to one invisible group or another. It can react against another drop drawn from an equally healthy human being. It can carry an identity that no face, name, nationality, or family legend reveals. The path toward Rh negative blood is not opened by tales of unusual ancestry. It is opened by failed rescues, endangered mothers, dying infants, and a laboratory dish in which red cells suddenly clump together, making visible, at last, the truth that had been present all along. Blood has names, even before humanity learns how to pronounce them. The breakthrough begins with a woman whose body has already suffered more than medicine can explain. In 1939, a patient enters the medical record not because she belongs to an ancient bloodline, not because anyone suspects a hidden human ancestry, but because an expected rescue fails in a frighteningly precise way. She has delivered a stillborn child. She requires blood. Her husband appears to be a suitable donor because, according to the blood group system physicians already trust, husband and wife are compatible, both belong to the same ABO group. The transfusion should not carry the classic danger Landsteiner's earlier discovery has taught doctors to avoid. Yet after receiving her husband's blood, the woman suffers a serious reaction. Something in his red cells has met something in her serum, and the result makes plain that the known map of blood is still missing a border. Two investigators, Philip Levine and Rufus Stetson, studied the case. When the woman's serum is tested against samples from other people of her own ABO group, it reacts with the red blood cells of a striking majority of them. That single observation carries an unsettling implication. Her husband's blood has not harmed her because it is from the wrong ABO category. It has harmed her because his cells bear another feature, one shared by many people but absent from her own blood. More disturbing still, the investigators infer that she has not simply acquired this reaction through the transfusion itself. Her pregnancy may already have exposed her to the unknown factor through the blood of her fetus. The child, inheriting something from the father, may have carried red blood cells different enough from the mother's to stimulate her immune response. In the ruin of that pregnancy and the failed attempt to save the mother, two mysteries suddenly touch. Unexplained transfusion reactions and the destruction of a newborn's blood may be different faces of the same hidden incompatibility. At the time, the condition seen in severely affected infants is known by a name that describes the aftermath rather than the cause, erythroblastosis fetalis. The term refers to immature red blood cells appearing in the fetus's circulation, as its body desperately attempts to replace cells being destroyed. Affected babies may develop intense jaundice, profound anemia, enlargement of the liver and spleen, or a terrible generalized swelling now recognized as hydrops fetalis. The most severely affected may die before birth, as Levine and Stetson's patient's child had. Doctors have encountered the illness before. They can see what it does. They do not yet understand why the mother's blood would attack the child's blood, or why one child in a family might survive while another is devastated. The 1939 case offers a mechanism the mother has been immunized against an inherited red cell factor, present in her baby and in her husband, but missing from herself. This is one of the quieter turning points in medical history. There is no grand expedition, no recovered tablet, no ancient tomb opened beneath torchlight. There is serum in a laboratory vessel and cells that clump when they should not. Yet the discovery reaches backward into countless lives. With one unusual case, losses that once appeared unrelated begin to align the stillborn child, the severely jaundiced newborn, the woman reacting to apparently compatible blood. The later pregnancies ending more disastrously than the first, a blood difference passed from parent to child and remembered by the immune system, can explain a pattern that has haunted families for centuries. It does not explain every stillbirth or every sick newborn. The evidence remains incomplete for most historical cases, and many disorders produce similar tragedy. But for the first time, the invisible enemy has left a fingerprint. Almost simultaneously, another line of investigation is moving toward the same territory from a different direction. Karl Landsteiner, now working in the United States, and Alexander Wiener are experimenting with red blood cells from rhesus macaques. By injecting those cells into rabbits, they produce an antibody that reacts not only with the monkey cells, but also with the red cells of a large majority of human beings. The reaction appears to reveal another widely shared human red cell characteristic beyond ABO. In 1940, Landsteiner and Wiener report this factor and name it for the rhesus monkey used in the experiment the Rh factor. The name will endure far longer than the original interpretation. Later work shows that the factor detected through the monkey experiment is not precisely identical to the clinically important human antigen involved in the pregnant patient's case. The historical naming is untidy, as scientific names often become once early discoveries are refined. By the time the distinction becomes clear, Rh has already entered medical language, blood bank practice, and public understanding. The important human marker becomes known as the D antigen within the Rh blood group system, while the original monkey-associated reaction is later recognized as belonging to a different but relatedly named system. Scholars of blood group history still untangle the sequence of discovery and terminology, because the clinical clue and the experimental clue emerged so close together and were initially treated as one. But for patients, the lasting result is simple and enormous. Medicine has identified a second major boundary in human blood. This boundary is different from the familiar ABO divide. A person with type A blood naturally has antibodies that can react against B cells. A person with type O blood can react against both A and B cells. Those dangerous antibodies often exist without an earlier transfusional pregnancy, making an ABO mismatch capable of producing a swift and violent crisis. The Rhd difference generally operates with a more tragic delay. If a person lacks the D antigen, there is not usually a naturally occurring anti-D army waiting in the blood from birth. The immune system first has to encounter D-positive red cells. That encounter may come through transfusion. For a woman carrying an Rhd-positive fetus, it may come when fetal cells pass into her bloodstream, particularly around delivery or other pregnancy-related events. Her body learns, it repairs, it may seem that the crisis has passed. Then, if a later fetus also carries the D antigen, the antibodies formed from the earlier encounter can cross the placenta and begin destroying the new child's red cells. The cruel timing helps explain why the condition confused generations of families and physicians. A first Rhd-positive infant born to an Rhd-negative mother may escape severe disease because the mother has not yet formed enough destructive antibodies during that pregnancy. The birth itself may be the moment that sensitizes her. A later child, genetically similar to the first, may face far greater danger. To those outside the laboratory, the sequence feels irrational. Why does the first baby live while the second becomes ill? Why can one mother bear several healthy children and another face recurrent loss? Why should blood from a husband, seemingly suitable and lovingly offered, cause harm after the couple has already lost a child once the Rhd antigen is understood? The pattern is no longer supernatural or arbitrary. It is immune memory, powerful and indifferent, operating exactly as it was shaped to operate against unfamiliar biological markers. Even when those markers belong to an unborn child, the discovery also reveals how misleading the everyday word negative can be. A person called Rh positive carries the D antigen on the surface of their red blood cells. A person called Rh negative lacks that antigen. The minus sign does not refer to weakness, contamination, inferior blood, or an abnormal emptiness. It does not indicate that every part of the Rh system is absent. The Rh blood group system contains several significant antigens, classically described as D, C, E, and E, with D becoming the most famous because of its powerful ability to provoke an immune response. In ordinary medical shorthand, the plus or minus written after A, B, a B, or O refers chiefly to the presence or absence of D. A negative means red cells carry the A antigen but do not display D. O positive means red cells carry neither A nor B, but do display D, a tiny symbol, attached to a familiar blood group letter. It begins to carry information that can determine which blood is safe in a transfusion and which pregnancy requires careful protection. In the years after the discovery, the new factor rapidly proves its importance. Blood banks begin to recognize why some patients react even when ABO-matched blood has been used. Obstetricians gain an explanation for many cases of hemolytic disease in newborns. Laboratory workers learn to look for anti-D antibodies in a mother's blood and to identify whether red cells carry the D antigen. The connection between maternal antibodies and infant disease becomes increasingly clear, especially through further advances in testing. In the mid-1940s, the antiglobulin test, often called the Coombs test, allows doctors to detect antibodies attached to red cells or circulating in serum with far greater precision. A newborn's damaged cells no longer speak only through jaundice and anemia. The antibodies coating them can be demonstrated. The ancient tragedy has become a detectable process. Detection, however, is not yet prevention. To know what is killing a fetus is not automatically to possess the means to stop it. For families in the early Rh era, a diagnosis can bring a severe kind of clarity. Mothers who have already become sensitized may be told that future pregnancies with Rhd-positive babies carry danger. Physicians begin developing ways to monitor affected pregnancies and to intervene in extreme cases, but the route is risky and limited. Exchange transfusion after birth, in which an infant's affected blood is gradually replaced with suitable donor blood, can save some newborns by reducing antibodies and damaged cells. Later, even more daring techniques will attempt transfusion before birth. Each advance is remarkable, but each begins only after danger exists. The deeper solution must be to prevent the mother's immune system from learning the D antigen in the first place, and that discovery still lies ahead. For now, the impact is intellectual as much as clinical. The Rh discovery forces medicine to accept that an ordinary pregnancy can be an immunological event, that the placenta does not form a perfect wall between two blood identities, and that inheritance can create danger not because a child carries a disease, but because a child carries a marker the mother lacks. It also makes the bloodline idea newly seductive. Here is an inherited trait, unevenly carried among people. Capable of making some pregnancies hazardous when positive and negative meet, it is easy, especially outside medicine, to transform that reality into a myth of incompatible peoples. But the discovery itself says nothing of separate species, mysterious ancestors, or forbidden crossings. It identifies a surface antigen. The danger occurs not because Rh-negative and Rh-positive people are fundamentally different kinds of human beings, but because the immune system responds to small differences with enormous force. That distinction will matter more and more as the decades pass. The very success of the medical discovery creates material for misunderstanding. Once a rare trait has a name, people can gather around it, Once it is linked to pregnancy loss, it acquires emotional weight. Once its distribution across populations begins to be measured, it appears to offer a trail into the distant past. Yet in the laboratory where the mystery first yields, the result is sober. There is no evidence of power in the negative blood, only absence of D. There is no evidence of conspiracy, only the long delay required before science possessed the instruments and concepts to see the cause. There is no proof that Rh negative blood comes from a lost race, only proof that inherited red cell differences can be medically decisive. A mother's tragic case has opened a new field of vision. The cells that reacted in her serum are no longer mute. Their clumping has given a signal a name. From then on, the minus sign can begin to mean something precise, not a curse written into blood, not an ancient secret guarded from the world, but the lack of one powerful antigen, harmless for most of daily life and profoundly important at the moment when one bloodstream enters another. Or when a child inherits a mark its mother does not share. Once a blood difference has a medical name, a deeper question appears almost immediately. What, exactly, creates it? The answer does not lie in the colour of blood, which remains the same vivid red whether a person is Rh positive or Rh negative. It does not lie in a separate organ, an unusual appearance, or a hidden substance circulating through a rare group of people. The answer lies in an instruction written into DNA, inside cells too small to be seen without magnification. On a chromosome shared by every human being, the Rhd antigen is not a mystical ingredient added to one bloodline and denied to another. It is a protein structure displayed on the membrane of red blood cells when the relevant genetic instructions allow it to be made. Where those instructions are absent, disrupted, altered, or expressed differently, the D antigen may not appear in the usual way. The result is the familiar negative sign. Seen from the scale of a birth certificate or a family legend, that sign can seem monumental. Seen from the scale of genetics, it is a variation in the biological machinery that builds the surface of a red cell. Human blood is crowded with such variation. The ABO letters are only the most familiar landmarks, and Rhd is only the most famous additional marker. Red cell surfaces carry hundreds of antigens belonging to many recognized blood group systems, most of which ordinary people never hear about unless transfusion becomes complicated, a pregnancy requires investigation, or a rare donor search begins. The Kell, KID, Duffy, MNS, and other systems each describe inherited molecular differences capable, under certain circumstances, of provoking immune responses. A person can walk through an entire lifetime carrying one of these unusual combinations without feeling any effect from it. The body is not a conscious reader of its blood group. It simply builds cells according to inherited instructions. Difficulty arises when blood bearing one set of markers is encountered by an immune system accustomed to another. Rhd became famous because it is particularly likely to provoke important antibodies, and because pregnancy provides a recurring natural setting in which that encounter can occur. Its fame does not make it supernatural, it makes it clinically consequential. The genetic machinery behind the Rh system is more intricate than the shorthand suggests. Two closely related genes, commonly called Rhd and Rhce, sit near each other on chromosome 1. They provide instructions associated with the Rh proteins carried in the membrane of red blood cells. The Rhce gene contributes to the C, E, and E antigen patterns, while the Rhd gene is principally responsible for the D antigen that determines whether most people are described as Rh positive or Rh negative. These genes are close relatives, shaped by duplication deep in evolutionary history, and their similarity helps explain why the Rh system contains not just simple presence or absence, but many variants, hybrids, and altered expressions. Most everyday conversations compress all that complexity into a plus or minus sign. In a hospital, that shorthand is extremely useful. In the story of human origins, however, the details matter, because they reveal how easily ordinary genetic processes can produce variation that later appears mysterious. Among many people of European ancestry who test as Rhd negative, the underlying cause is relatively straightforward. The Rhd gene is absent from both copies of chromosome 1 inherited from the parents. Everyone carries two versions of most autosomal genetic regions, one inherited from the mother and one from the father. If a person receives functioning Rhd instructions from at least one parent in the common pattern, their red blood cells usually carry the D antigen and they are typed as Rh positive. If they inherit forms in which the Rhd gene is absent on both sides, their cells do not show the D antigen in standard testing, and they are typed as Rh negative. This is not an impossible gap in the human genome. It is not evidence that something essential was removed by an unknown event. Gene deletion is one of the ordinary ways genomes change over time. Sections of DNA can be copied, lost, rearranged, or blended through errors during reproduction, and if individuals carrying a change survive and have children, that variation may continue through a population. The word deletion can sound alarming, especially when attached to a trait associated with infant danger. But genes are not a sacred checklist in which every absence signals damage or degeneration. Some deleted genes are harmful. Some have little apparent effect except in particular environments. Some may even be helpful under certain conditions. In the case of common Rhd negative blood, the absence of Rhd does not prevent red blood cells from carrying oxygen, circulating through vessels, or performing the daily labour that sustains life. Rh negative people do not require special diets to compensate for a missing antigen. They do not possess a different form of hemoglobin because of it. They are not medically defective. The consequence becomes important chiefly when their immune system encounters D positive blood. whether through transfusion or pregnancy. One might compare it not to a missing engine in a vehicle, but to a different flag on the surface of a ship. Irrelevant while sailing alone, highly significant when another force is trained to recognize flags. Even the clean division between positive and negative begins to blur when geneticists and transfusion specialists look more closely. Not every person described as Rh negative carries the same genetic arrangement, and not every Rh positive result represents the same strength or form of D antigen. In several populations, including many people of African ancestry, forms of the Rhd gene occur that produce altered or weak expression of the D antigen. Some red cells display only portions of the usual D structure, while others display it in reduced quantity. These are known through categories such as weak D or partial D, though the exact classifications can be medically complex. A person may test one way in a routine setting and require more careful molecular typing when pregnancy or transfusion decisions are involved. A surprising detail lies here. Two people whose hospital records both contain a plus sign may not carry identical versions of the Rhd antigen, and two people whose blood appears negative in one testing context may not have precisely the same genetic story beneath that result. This complexity is the opposite of what a hidden bloodline myth requires. Myths thrive on sharp borders, one ancient lineage versus another, special people versus ordinary people, a single lost origin explaining all carriers of a rare trait. Genetics presents a less theatrical but far more interesting reality. Rhd negative status can arise through different molecular routes in different human populations. The deletion frequently found among Europeans is not simply copied everywhere in the world in the same pattern. In other ancestry groups, negative or unusual Rh typings may reflect altered forms of the gene. Hybrid genes formed through recombination between Rhd and Rhce, or expressions that standard tests must interpret carefully. If all Rh negative people descended from a single mysterious source marked by one extraordinary biological event, the genetics ought to offer a clean signature. Instead, it reveals the familiar texture of human evolution shared ancestry, repeated variation, migrations, genetic mixing, and local histories that preserve different versions of similar outcomes. There is another reason the D antigen inspires speculation. It seems strangely unnecessary until it suddenly matters. A person lacking it functions normally, yet a person possessing it can trigger a dangerous reaction in someone who lacks it. Why does humanity carry such a system at all? Here caution is essential. Rh proteins are embedded in the red cell membrane and are related to the transport of substances across cell membranes. The Rh complex appears important to the stability and function of red cells, but the specific evolutionary story of the common D positive and D negative forms is not reduced to one neat explanation. Scientists can identify genes, compare frequencies, examine variants, and model inheritance. Establishing exactly why particular forms rose or persisted across prehistory is more difficult. Selection may have mattered in some settings. Chance and population history may have mattered enormously. The evidence remains incomplete, especially because the relevant genetic events occurred long before written records and in populations that have since moved, merged, expanded, and disappeared. What is not uncertain is the basic inheritance. An Rh negative mother does not give birth to an Rh positive child because some mysterious foreign quality has entered the family. She can do so because the father contributes an Rhd bearing chromosome and the child inherits it. An Rh positive person can carry one copy associated with D expression and one without, meaning two Rh positive parents can, under the right inherited combination, have an Rh negative child. The trait can pass quietly through generations and appear where a family does not expect it, which in earlier ages might easily have fed rumours about parentage or unusual ancestry. In reality, recessive inheritance often works in just this hidden way. A characteristic may be carried invisibly in people who do not display it in the common test result, only to appear when two inherited paths meet in a child. This subtle inheritance is one reason blood groups have played complicated roles in human imagination. They're genuinely ancestral, in the sense that they pass from parents to children and carry traces of population history. Yet they are poor tools for declaring a person's entire origin, identity, or destiny. A blood group variant is one thread among billions of positions in the genome. It can offer researchers information when studied across large numbers of people. Alongside other genetic evidence and archaeological context, it cannot, alone, announce that an individual belongs to an untouched ancient tribe, much less a separate form of humanity. Many unrelated people can share a trait because their distant ancestors carried it, because populations mixed, or because similar genetic outcomes emerged through related molecular processes. The minus sign can be meaningful without becoming a passport to a secret past. The misunderstanding deepens because Rhd negative blood is often described as though it contains something rare and powerful. In fact, with respect to the D antigen, the defining feature is the absence of a surface marker that most people carry. This makes Rhd negative red blood cells valuable in certain transfusion situations, particularly because they cannot expose a recipient to the D antigen that is not present on them. But usefulness in medicine is not proof of heightened human ability. A donor can be urgently needed because their blood is compatible with a vulnerable recipient, not because it contains superior properties. Similarly, a mother may need anti-D protection not because her body is weaker than others, but because a normal immune response can be dangerous when directed at fetal red cells carrying an antigen she lacks. At the extreme edge of the Rh story lies a condition far rarer and fundamentally different from ordinary Rhd negative status, Rh null blood, sometimes sensationally called golden blood. People with Rh null lack all Rh antigens on their red cells, not merely the D antigen used in the everyday positive-negative designation. Their blood is extraordinarily valuable for certain rare transfusion needs. But the condition may also be associated with abnormalities in red cell shape and chronic hemolytic anemia. It is not a hidden upgraded version of Rh-negative blood. It is a medically important and exceptionally rare phenotype. The distinction is frequently lost in popular retellings, where ordinary Rh-negative people are blended with the rarest blood cases to make the entire subject seem more extraordinary than it is. The real story is already extraordinary enough. Human cells carry an elaborate molecular identity system, and a tiny fraction of that system can shape pregnancy, transfusion, and survival. By the time genetics begins to reveal the architecture beneath the D antigen, the old mystery has changed form. Medicine no longer needs to wonder whether Rh-negative blood comes from outside the ordinary human inheritance system. It can trace the trait to genes, observe how parents pass it to children, identify variants, and distinguish simple D absence from more complex Rh expressions. The conspiracy suggested by popular imagination begins to weaken under the microscope. There is no missing chapter that requires an alien donor or a vanished superhuman population. There is a genetic region subject to the same forces that have shaped every other part of the human genome mutation: recombination, inheritance, chance, survival, and movement across landscapes. Yet one question remains strong enough to carry the story forward. If Rhd-negative blood arises through ordinary genetics, why is it so uncommon in many places and so much more frequent in others? A gene can explain how a trait is inherited. It does not automatically explain the map it makes across the world. That map, once drawn, will become the fertile ground on which ancient lineage theories grow. But before myth claims it, the pattern must be seen for what it is. Not a message deliberately hidden in blood, but a surviving trace of how human populations have wandered, separated, endured, and carried their smallest inherited differences with them. A gene becomes a mystery to the wider world when it is placed on a map, on a laboratory report. Rh-negative blood is simply a result. In a family, it is an inherited detail that may matter during pregnancy or transfusion. But when researchers begin counting how often that result appears among different populations, the pattern seems to sharpen into something larger. Rhd-negative blood does not occur at the same frequency everywhere. In much of East Asia, it is uncommon. Across many African populations, the simple European-style Rhd-negative pattern is also relatively uncommon, though the Rh system there contains important diversity and altered variants of its own. In Europe, the negative phenotype becomes markedly more frequent, and in parts of Western Europe, especially among Basque populations living around the Western Pyrenees, reported frequencies have long drawn attention for being unusually high. A tiny molecular absence, almost meaningless to the eye, begins to look like a trail across continents. This is the point where the subject acquires much of its modern aura. Numbers on population surveys can feel like the outlines of an ancient secret. Why should one inherited trait gather in particular places? Why should people separated by mountains, language, seas, or migrations carry different proportions of the same blood feature? Such questions are legitimate. Population genetics is built from them. Human beings do not spread evenly across the earth and exchange genes without interruption. They move in waves, settle in valleys, cross straits, become isolated on islands, expand after famine or disease, absorb neighbouring communities, and sometimes pass through centuries in which a small number of ancestors leave an unusually large imprint on the future. Blood group distributions preserve traces of those events, but a trace is not the same as a revelation. It must be interpreted cautiously, because a map of modern blood frequencies is not a photograph of the Ice Age, nor a simple family tree of ancient peoples. In broad terms, Rhd-negative blood is substantially more frequent among people with European ancestry than in many other large regional populations. Depending on the population studied and the definitions used, roughly 1 person in 6 among many European-derived groups may be Rhd-negative, while the proportion is often much lower in East Asian populations and lower in many populations of sub-Saharan African ancestry when ordinary D-negative typing is considered. Such comparisons must be handled carefully. African populations, in particular, hold extensive genetic variation within the Rh system, including Rhd variants that complicate simple categories of positive and negative. Modern nations also contain immense mixtures of ancestry, migration, and local history. No country is one genetic block, and no blood frequency describes every family within it, yet the broad pattern is real enough to demand explanation. The D-negative phenotype common in Europe appears at much lower rates across many other regions of the world. To a person seeking a dramatic answer, the unevenness can seem suspicious. If Rh-negative blood was simply an ordinary variation, why would it not be evenly scattered everywhere? But ordinary genetic variations are rarely scattered evenly. Skin pigmentation, lactose digestion, resistance or vulnerability to certain diseases, the frequency of particular enzymes, and countless harmless markers all vary from population to population. Some differences reflect adaptation to local environments. Others reflect who happen to survive and leave descendants. Still others are shaped by isolation, marriage patterns, epidemics, population replacement, or simple statistical chance magnified over many generations. The human species has always been one connected, interbreeding species, but not one perfectly mixed population. For most of prehistory, people lived in communities whose marriage networks and travel distances were far smaller than those of the modern world. A rare trait carried by the founders of such a community might become locally common, even if it offered no special power at all. The visual temptation is strongest in Western Europe. The Atlantic-facing edge of the continent has often appeared, in the imagination, as a kind of refuge, a place of green mountains, abrupt coasts, deep caves, old languages, and populations thought to preserve echoes of earlier Europe. Here, near the Bay of Biscay and in the folds of the Pyrenees, live the Basque people, whose language, Euskara, is unrelated to the Indo-European languages now spoken across most of Europe. The Basque region is not an isolated lost kingdom sealed away from history. Its people have traded, fought, married, migrated, and interacted with neighbours for millennia. Yet the survival of a non-Indo-European language in Western Europe is genuinely remarkable. When early blood group studies also reported relatively high Rh negative frequencies among Basque populations, two real observations were placed beside one another: a rare linguistic survival and an unusual blood group distribution. From that pairing, the imagination needed little encouragement. Soon the Basques were cast in stories as relics of Cro-Magnon Europe, direct descendants of Ice Age populations, survivors of vanished civilizations, or keepers of an ancient bloodline different from the peoples surrounding them. Raw negative blood became one of the props used to support those narratives. If a population has a distinctive language and a relatively high frequency of a medically significant blood trait, perhaps, the argument goes, it must descend from some exceptionally ancient group. But population history is not so clean. Modern genetic research has refined older romantic pictures. Basque ancestry does show important continuity with ancient Western European populations, and later population history in the region produced distinctiveness relative to some neighbours. Yet the Basques are not a biological fossil, preserved unchanged from the Paleolithic. Like all living populations, they carry layers of ancestry and evidence of contact, mixture, isolation, and change. Their story is fascinating precisely because it is human, not because it stands outside humanity. Early population surveys helped turn Rh negative blood into a subject of public fascination, partly because blood groups were among the first genetic markers that could be widely compared before modern DNA sequencing existed. Researchers collected samples from towns, regions, military recruits, isolated villages, and communities with known ancestry. Blood types became a way of estimating biological relationships among populations, though an imperfect one. If two communities shared similar frequencies of certain blood markers, perhaps they shared ancestry or contact. If they differed, perhaps their histories had diverged. Blood group maps were useful, but limited. A single marker can be heavily influenced by chance. Different historical pathways can produce similar frequencies. Later migration can reshape a pattern. A population can share a high rate of one blood feature with another, while differing strongly across the rest of the genome. This is the difference between a clue and a conclusion, a difference popular retellings often erase. The uneven map also contains traps of language. People often say that negative blood is rare, which is true globally and in many local contexts, but rarity depends on where one is standing. In one community, an Rh negative patient may be one among a substantial minority. In another, suitable Rh negative blood may be extremely difficult to obtain quickly. A trait can be uncommon across the world while still familiar within a particular regional ancestry group. This matters in hospitals. Blood banks must anticipate the needs of the actual populations they serve, and changing migration patterns can create demands for donor matches that were once rarely encountered in a given place. It also matters in storytelling. When a feature is rare enough to feel special but common enough for millions of people to identify with it, it creates a ready-made audience for claims of secret inheritance. There is an additional complication. The frequency of Rh negative typing does not reveal only ancient population movement. It is also connected to how blood is tested and how particular genetic variants are classified. In one population, apparent D-negative status may commonly result from deletion of the Rhd gene. In another, molecular variants may lead to weaker or partial antigen expression, sometimes presenting challenges for older testing methods or for the interpretation of transfusion risk. The world map is therefore not a single clean distribution of one identical mutation. It is a map of phenotypes shaped by multiple genetic backgrounds and different local histories. This is a lesser-known detail, and it undermines the idea that every Rh-negative person belongs to one hidden global lineage. Similar clinical labels may sit above different genetic arrangements. Even within Europe, there is no sharp frontier where one mysterious bloodline ends and another begins. Frequencies rise and fall gradually across regions, influenced by past population structure and later movement. An individual born far from the Pyrenees may carry Rh-negative blood through ancestry spread across Western or Northern Europe, through mixed family histories, or through patterns too ordinary ever to have been recorded. Conversely, most people in any population associated with high Rh-negative frequency are not defined by that trait alone. A Basque person may be Rh positive. An East Asian person, though less commonly, may be Rh negative. Blood does not obey cultural borders, and no living community is reducible to one antigen. Still, there is something moving in the thought that blood frequencies carry faint marks of journeys no chronicler recorded. Long before passports, borders, and surnames, small groups made decisions that shaped the living world. They followed herds across cold landscapes, sheltered along coasts, retreated from ice, crossed river valleys, remained in mountain basins, or expanded through newly fertile plains. A couple carrying a particular genetic variant had children. Perhaps those children found partners within a relatively small group. Perhaps war or famine narrowed the population. Perhaps centuries later the variant remained in a higher proportion than elsewhere. No one in that chain needed to know the trait existed. No one needed to conceal it. Inheritance preserved it without story, while history supplied the conditions under which it became common or rare. The medical cost makes the persistence of the RHD negative phenotype seem, at first, even more perplexing. If an RHD negative mother can become sensitized and lose RHD positive children in later pregnancies, why did the trait not simply disappear under the pressure of natural selection? The answer is not yet fully settled, but the problem is less impossible than it sounds. Not every RHD negative woman has children with an RHD positive father. Not every such pregnancy causes sensitization. Not every sensitized pregnancy ends in death. The cost, while real and sometimes devastating, is uneven. A trait may persist if its disadvantage is limited, if chance keeps it common in a population, if there were unknown balancing influences, or if demographic history overwhelms selection in particular times and places. Scholars still debate the details of our evolution, and no single explanation fully accounts for the entire global distribution. But persistence does not require a hidden origin. Countless genetic traits with costs remain in human populations because evolution is not a perfect machine for eliminating every vulnerability. The map of Rh negative blood is therefore both less sensational and more haunting than legend suggests. It does not point to an alien intrusion or a secret caste of ancient survivors. It points to human groups moving unevenly through deep time, carrying variations whose future meaning they could never foresee. It points to mountain communities and coastal refuges, to founder effects and marriages, to the terrible randomness of who survives an epidemic, a famine, a winter, or a war. It points to mothers who inherited a rare blood pattern without knowing it, and to children whose lives once depended on a compatibility no one could test. By the modern era, those scattered frequencies have become easy to look up, easy to compare, and dangerously easy to overinterpret. One region, above all, will come to stand at the centre of the popular mystery of the Basque country, where language, landscape, ancestry, and blood appear to converge into the shape of an unsolved riddle. The real history there is not a proof of forbidden origins. It is the story of how isolation and continuity can preserve unusual signals, and how quickly an unusual signal can be transformed into a legend of people set apart. In the steep green valleys at the western end of the Pyrenees, the mystery seems, for a moment, to acquire a homeland. The mountains do not form an impenetrable wall here. Roads and passes have always led through them, and the sea has long carried fishermen, traders, raiders, and emigrants outward and home again. Yet the landscape encourages the eye to imagine survival. Stone farmhouses sit among wet hills. Rivers descend toward the Bay of Biscay. Caves in the wider region contain traces of people who lived during the last ice age, when reindeer and bison moved through a colder Europe and painted animals appeared on rock in the darkness underground. In this corner of Spain and France, a people speak a language that does not descend from Latin, Celtic, Germanic, or any other surviving Indo-European branch. It is called Euskara, to outsiders. Its survival has often seemed like a voice carried intact from a Europe that vanished before history began. Add a comparatively high frequency of Rh negative blood, and the ingredients for legend gather almost by themselves. The Basques have been made to carry many stories that did not begin with them. They have been described as Europe's first people, as untouched descendants of Stone Age hunters, as refugees from Atlantis, as the remnant of a civilization erased by flood or conquest, and, in more recent fringe accounts, as the keepers of a bloodline fundamentally unlike that of their neighbours. In these tales, negative blood is treated as a biological seal stamped onto an ancient identity. The reasoning appears seductive because it combines genuine facts. Euskara really is a language isolate, meaning that no demonstrated living linguistic relatives survive. Basque communities really did retain a strong cultural identity through centuries of Roman influence, medieval kingdoms, shifting borders, and industrial modernity. Older blood group studies really did draw attention to elevated Rhd negative frequencies among Basques. But genuine facts do not automatically support the larger story constructed around them. A surviving language is not proof that a population has remained genetically sealed since the Ice Age. A high frequency of one blood marker is not evidence of a separate origin, and a mountain landscape, however ancient it feels, is not a wall against every movement of people. For much of the 20th century, scientists had relatively few tools with which to investigate deep ancestry. Before whole genomes and ancient DNA could be read, blood groups were valuable clues. Researchers measured ABO and Rh frequencies, along with a growing collection of other biological markers, and compared one population with another. Basque samples appeared distinctive in several respects, particularly their notably high proportion of Rh negative individuals in some surveys. Reported percentages differed by location, sampling, and era, but the pattern was striking enough to become famous. It seemed to fit the language a population unusual in speech, also unusual in blood. In an age eager to sort European prehistory into ancient races and surviving types, the conclusion seemed almost irresistible. The Basques were often portrayed as the direct, minimally altered descendants of the people who occupied southwestern Europe before later farmers, metalworkers, Celts. Romans and other newcomers changed the continent. The old picture contained a fragment of truth but made it far too simple. The Basque region does preserve a notable degree of historical distinctiveness, and its population has experienced periods of relative isolation that can help maintain particular genetic patterns. Yet prehistoric Europe was never a stage upon which one pure people stood still while every other population arrived around them. Ancient DNA has revealed repeated transformations across the continent. The hunter-gatherers who lived after the Ice Age were joined and substantially reshaped by farmers whose ancestors came from Anatolia thousands of years later. During the Bronze Age, additional ancestry associated with steppe pastoralist expansions spread widely across Europe. Iberia, including the wider region from which later Basque communities emerged, participated in these enormous demographic changes. The ancestors of modern Basques were not untouched spectators outside those movements. They were formed within them. What makes their history interesting is not that they somehow escaped prehistory, but that later patterns of contact and isolation helped preserve a distinct combination after major ancestral layers were already in place. Genetic studies comparing modern Basques with ancient individuals from the Iberian Peninsula suggest substantial continuity with Iron Age populations in northern Iberia, followed by reduced mixing with some later incoming groups relative to surrounding communities. In broad terms, their distinctiveness appears to have sharpened in historical rather than purely Paleolithic time. They are not fossils walking through the present. They are descendants of ancient Iberian populations who, after sharing in the great prehistoric migrations that shaped Western Europe, maintained a language and a degree of genetic continuity through later centuries when neighbouring groups experienced different mixtures. The evidence remains incomplete, as ancient DNA coverage is always uneven, and every new sample can refine the picture. But it decisively weakens the romantic idea of a bloodline sealed unchanged since the caves were painted. Language makes the story feel older than genetics alone can prove. Euskara survives where earlier non-Indo-European languages across much of Europe disappeared, replaced by tongues connected to expanding farmers, pastoralists, empires, kingdoms, and states. Its persistence is remarkable. It may retain connections to languages once spoken more widely in the region, ancient inscriptions associated with Aquitanian, found north of the Pyrenees under Roman rule, contain personal and divine names that show clear links to Basque. That is a rare and tangible glimpse of linguistic continuity reaching back into antiquity. Yet languages and genes do not travel in identical ways. A community can adopt the language of a minority elite, preserve an old language while absorbing new families, or lose its language without undergoing wholesale population replacement. Euskara tells us that cultural continuity was unusually strong. It does not give a simple measure of how much every Basque ancestor differed biologically from every neighbouring ancestor. The blood marker follows the same rule. A high local frequency of Rhd negative blood can emerge and persist through population structure, not through separation into another kind of humanity. Imagine a region in which the D-negative genetic arrangement is already present at a meaningful frequency, if communities marry mainly within local networks over long periods, and if chance events leave certain family lines especially numerous. The frequency can remain high or even become higher than in surrounding areas. Mountains, political boundaries, language, local inheritance customs, village ties, and social identity can all reduce gene flow without eliminating it. A rare feature does not require an origin in a rare species. It requires carriers and a history in which their descendants remain numerous enough for the feature to endure. Even the famed Basque art negative rate demands restraint. It is sometimes repeated online as a single dramatic number, as though every Basque village and every study returned the same result. They do not. Populations are not laboratory jars. Sampling varies. Rural and urban histories differ. Centuries of migration have moved Basque descendants to the Americas and brought others into Basque towns. Within the region, individuals have differing family histories, and Rh-negative status is only one inherited trait among an enormous number. The notable frequency is meaningful at the population level. It says almost nothing decisive about any individual's deep origin. A Basque person who is Rh positive does not become less Basque. A non-Basque person who is Rh negative does not acquire an ancestral claim to Euskara-speaking valleys. Biology and belonging overlap in history, but they are not interchangeable. There is a more uncomfortable reason the idea of the Basques as a separate blood people has been so persistent. During the 19th and early 20th centuries, European scholarship and politics were saturated with efforts to classify human communities into ranked races, ancient types, and supposedly pure lineages. Measurements of skulls, descriptions of faces, language families, and later blood frequencies were often pulled into arguments about identity and difference. Some researchers approached the subject carefully. Others treated limited evidence as proof of sweeping racial narratives. The Basques, because of their language and location, became a favoured object for such theories. In later popular culture, the vocabulary changes. Instead of racial anthropology, one hears of star ancestry, secret DNA, forbidden blood, or authorities hiding the real origin of Rh-negative people. But the underlying desire remains similar: to turn a complicated living population into evidence of a sharp biological divide. The irony is that Basque history needs no invented divide to be extraordinary. A community can preserve a language through waves of political and economic pressure, only through countless acts of daily continuity, children learning words at home, shepherds naming hills and weather, songs crossing generations, legal customs maintained, seafarers leaving and returning, villages adapting without dissolving, Roman roads passed through the region. Christianity transformed religious life. Medieval conflict drew boundaries and alliances across the mountains. Basque sailors became central to Atlantic fishing and whaling, reaching distant northern waters centuries before modern industrial fleets. Later, Basque migrants helped shape communities across the Americas. This is not the record of people untouched by the world. It is the record of people repeatedly engaged with the world while preserving something unusually durable within it. One lesser-known twist in the story lies in how easily a medical characteristic can become entangled with national or regional pride. A population told for generations that its blood is distinctive may begin to view an ordinary blood test result as confirmation of a deeper identity. For some, that can be benign, a point of curiosity alongside language and family origin. For others, it can attract stories of superiority, purity, or biological isolation that history does not support. Our negative blood does not belong to the Basques, and the Basques are not defined by Or's negative blood. The phenotype exists across many populations, and within the Basque country, it is distributed among ordinary families living ordinary lives, important in the same settings where it matters everywhere else: transfusions, blood donation, and pregnancy. The true link between the Basque region and Arge negative blood is therefore neither meaningless nor mystical. It is a real population pattern, likely shaped by ancestry, local continuity, and periods of reduced gene flow. It offers one clue among many to the complicated history of Western Europe, but it cannot bear the weight placed upon it by claims of a lost race or an inhuman origin. If anything, the Basque example demonstrates how human history works at its most subtle. Traits survive not because they announce themselves, but because people survive. Languages persist not because time stops, but because communities continue speaking them through change. A blood group frequency may last for centuries not because an ancient secret is protected by silence, but because the ordinary decisions of marriage, family, geography, and belonging leave marks that outlive every individual involved. Beyond the Pyrenees, the same forces have worked on every human population. Small groups separated from others, expanded, contracted, met new neighbours, endured harsh climates, followed new foods, carried alleles into distant places, and left some variations clustered more strongly than others. To understand why Rh negative blood appears where it does, the story must move deeper than a single people, however compelling their history may be. It must return to the prehistoric roads along which genes travelled without maps, the refuges of the Ice Age, the arrival of farmers, the narrowing of communities, and the simple power of chance when only a small number of ancestors stand at the beginning of many descendants. Long before anyone spoke Euskara, before any boundary separated France from Spain, before blood could be drawn into glass tubes and placed beneath a laboratory lamp, the landscapes of Europe repeatedly changed their invitation to human life. Ice advanced and retreated. Forest opened into steppe and closed again into woodland. Coastlines shifted as sea levels fell and rose. River valleys widened, narrowed, froze, flooded, and offered passage in one generation where they had blocked it in another. In such a world, human ancestry did not flow like ink evenly spilled across a map. It moved in pulses. A group followed game southward as cold deepened. Another survived for generations near a coast rich in shellfish and fish. A few families crossed into a valley newly exposed by retreating ice. Some settlements flourished, while others ended without descendants, leaving their genetic signatures buried alongside flint flakes, hearth ash, and bones gnawed clean in the dark. Every blood trait carried in living people today has passed through those narrowing corridors of survival, including the genetic arrangements that can produce Rh negative blood. The last great glacial period transformed much of Europe into a severe and uneven world. At its coldest phases, enormous ice sheets covered northern regions, while farther south, broad tundra and steppe stretched beneath bitter winds. Humans did not simply vanish from the continent, but populations contracted into more favourable areas, often described as refugia, where climate, terrain, and food made continued life possible. Southwestern Europe, including portions of Iberia and the region approaching the Pyrenees, became one of several places where people endured through the harshest conditions. Caves, rock shelters, river corridors, and coastlines offered protection and resources. The archaeological record preserves magnificent signs of their presence tools shaped from stone and bone, animal remains, pigments, ornaments, and painted chambers where horses, bison, ibex, and deer move across walls under flickering light. Those images reveal imagination and skill. They do not reveal blood types. No Ice Age handprint on a cave wall tells us whether the person who left it carried an Rh-D deletion or displayed the D antigen on red cells. The evidence remains incomplete, and modern Rh frequencies cannot simply be projected backward onto the artists of deep prehistory. Still, refugees matter because they create the conditions in which chance can become history. When a large, widely mixed population shrinks into scattered pockets, only a portion of its earlier genetic variation continues forward in each pocket. Suppose a blood group variant is uncommon across a broad population before a climatic crisis. If, by accident, it occurs in several of the families who survive in one refuge, its local frequency may become far higher than it had been before. If it is absent among another surviving group, that group's descendants may remain almost entirely without it. Nothing about the variant needs to improve hunting success, protect against winter, or grant special resistance. It may simply have been present in the right survivors, or absent from the wrong ones. This is the quiet force of genetic drift, the power of chance operating through small numbers. repeated generation after generation until a feature begins to appear characteristic of a region or a people. The idea is easier to grasp at the edge of a catastrophe. Imagine a storm-struck island inhabited by 1,000 people, of whom 50 carry an unusual inherited trait. If famine, disease, or evacuation leaves only 30 survivors, and by chance 10 of those survivors carry the trait, the next generation begins with a frequency vastly higher than before. No advantage has been proved. No secret bloodline has emerged. A narrow gate is simply selected, partly by accident. Which versions of human variation continue onward? Prehistoric life repeatedly created such gates, though usually less suddenly and over longer spans: glacial cold, local hunger, epidemics, dangerous crossings, exhausted prey, social conflict, and the isolation of small groups far from possible partners. Human beings are not only descendants of those who are best adapted in every respect, they are descendants of those who happen to live long enough, meet one another, and have surviving children. When warmer conditions returned after the Ice Age, people expanded outward again, valleys opened, forests spread into lands once dominated by open cold plains, animals shifted their ranges, demanding new hunting strategies and new movement. Human groups carried their inherited variations with them as they recolonized landscapes. A genetic marker that had become common in a southwestern refuge might then spread into neighboring regions, not because it was essential to the expansion, but because the people making the expansion happened to carry it. Such expansions can create geographic gradients, regions where a trait is more frequent near a source population and gradually less frequent farther away, especially if later movement is limited. In older interpretations, high Rh negative frequency in Western Europe was sometimes connected strongly to the descendants of Ice Age refugees, including romantic associations with the ancestors of the Basques. That possibility cannot be dismissed as though refugial population history meant nothing, but neither can it be treated as a complete answer. Later migrations profoundly reshaped Europe, and current evidence does not allow a simple statement that Rh negative blood is a direct badge of Paleolithic survivors. Thousands of years after glacial cold loosened its grip, another transformation altered the ancestry of the continent. Farming communities, ultimately tracing much of their origin to Anatolia and the Aegean region, expanded westward into Europe. They carried wheat, barley, livestock, polished tools, settlement patterns, and new ways of living on the land. Their movement was not merely the transfer of ideas from one untouched population to another. Ancient DNA shows that people moved as well, interbreeding with local hunter-gatherers in patterns that differed across regions and through time. In Iberia, as elsewhere, the genetic landscape became a mixture of earlier inhabitants and incoming farmers. Millennia later, during the Bronze Age, ancestry connected with populations from the Eurasian steppe spread widely through Western Europe, again altering the populations of the peninsula. Any inherited blood variant found among modern Europeans has moved through this layered world. Its present frequency may reflect several stages of history, not one dramatic origin. These changes help explain why a single marker can mislead when made to stand for an entire ancestry. An Rh negative person in Western Europe may carry a form of the Rhd region, whose deeper distribution was affected by prehistoric population events, but the rest of that person's genome is a vast mosaic of ancestral paths. The same is true of a population with an unusually high frequency of the trait. One marker can persist or become elevated even as most of the ancestry around it is reshaped by migration and mixing. The survival of an allele is not the survival of an unchanged people. A thread from an older fabric can be woven into many later garments and remain visible only because researchers know where to look. The founder effect makes this still more striking. A population does not need to pass through a continent-wide disaster for an unusual trait to become common. Sometimes a small number of people establish a new community, whether on an island, in a mountain valley, along a remote coast, or beyond a frontier. Their descendants inherit only the genetic range those founders carried. If one or more founders happen to possess a rare variant, that variant can become surprisingly frequent in the resulting community. This pattern appears throughout human genetics and in certain inherited disorders. It is not a mark of otherness. It is often a mark of isolation and growth from small beginnings. A fishing settlement, a pastoral valley, or a religiously or linguistically bounded community can preserve a genetic frequency that looks startling when viewed centuries later, even though it began with no mystery greater than a handful of families and the ordinary inheritance of children. For Rh negative blood, founder effects and drift offer a powerful framework because the trait is mostly silent in daily life. Before modern obstetrics, such incompatibility could impose serious reproductive costs in some families, but the trait did not prevent most carriers from reaching adulthood, working, migrating, marrying, and reproducing. An Rh negative man does not face the pregnancy-related immune problem in his own body, though he may father Rh positive children with an Rh negative partner depending on his genes. An Rh negative woman is at risk of sensitization only in particular reproductive pairings and circumstances, and even then, severe outcomes are not guaranteed. If a community contains many Rh negative men and women, Rh negative couples can have Rh negative children without the de-incompatibility at the centre of the danger. The disadvantage is real, but it is not absolute. In a small or somewhat isolated population, drift can preserve a trait despite a cost that appears puzzling when viewed from the outside. Some researchers have also asked whether RHD status may have been influenced by forms of natural selection beyond hemolytic disease in newborns. Certain studies have explored associations between Rh status and responses to infections or other biological pressures, but such questions are complex, findings can vary, and no accepted explanation has demonstrated a single ancient advantage responsible for the global map of Rh-negative blood. The temptation is always to demand that every persistent trait must have provided an advantage. Evolution does not work so neatly. A trait can survive because its disadvantages are limited, because any advantages are context-dependent or unknown, because it travels alongside other ancestry, or because random changes in small populations overpower weak selective pressures. No single explanation fully accounts for the history of Rhd negative frequencies across the world. And honest uncertainty is not a missing piece that conspiracy can automatically fill. There is a human dimension to these genetic mechanisms that their dry terms often conceal. A bottleneck is not simply a narrowing line on a population graph. It is winter hunger, a failed harvest, a disease sweeping through households, a coastline swallowed by rising water, a violence that scatters survivors, or a migration undertaken because remaining in place has become impossible. A founder effect is not merely an equation, it is a boat arriving on an island. Several families making homes in an upland valley. A group leaving conflict behind and building a village where their children marry one another's children. Isolation is not silence or purity. It is distance walked in bad weather. Mountain passes closed by snow, language shaping trust and marriage, work keeping communities close to land and sea, and the simple fact that until very recently most people spent their lives near where they were born. Through all these movements, blood travels without announcing itself. A mother nursing an infant in a cave refuge has no knowledge of antigens. A farmer clearing Iberian soil has no reason to imagine that one inherited detail within his family will suddenly be measured in a modern hospital. A Bronze Age traveller crossing into new settlements does not know which cellular markers will continue in descendants after languages, gods, tools, and borders have all changed. Yet each life carries genetic possibilities into the future. The Rhd gene may be present, absent, or altered. The child may inherit one version or another. Across generations, small frequencies rise and fall, not with the clarity of an intentional message, but with the accumulated force of ordinary survival. This is where the rare bloodline legend brushes against something true and then turns away from it. A negative blood does connect a living people to ancient events, but not uniquely, and not in the form of a secret ancestry. Every gene does this. Every human body is an archive written by migrations, bottlenecks, meetings, separations, illnesses, births, and chance. Rh-negative status attracts attention because it is medically important and unevenly distributed, and because its absence can be named with a stark minus sign. But the ancient road leading to that sign is not marked by forbidden crossings between incompatible kinds of humans. It is marked by the same difficult roads all ancestry has taken through cold refugees and warmer valleys, through expanding villages and fading camps, through strangers becoming families and families becoming populations. The wonder is not that Rh-negative people stand apart from humanity. The wonder is that something so minute can survive such distances in time. A missing antigen on a red cell, inherited through people who never knew it existed, passes through ice, migration, settlement, mixture, birth, and loss until it appears on a modern medical form. There, at last, it becomes visible, and once visible, it leads not to an alien past, but toward an even more intimate consequence. The moment that inherited variation meets pregnancy, and a mother's immune system begins to remember the blood of her own child. The ancient journeys of genes remain abstract until they arrive in the life of a particular family. A woman does not experience population frequencies or founder effects when she learns she's pregnant. She experiences the small physical signs of a body changing its priorities: exhaustion, hunger, nausea, the first uncertain movement that later becomes unmistakable. Her child is not a genetic diagram, but a presence growing within her, nourished through an organ of extraordinary delicacy. The placenta joins mother and fetus closely enough to sustain new life, passing oxygen and nutrients across an intimate boundary, carrying waste away, releasing hormones that reshape the mother's body around the demands of pregnancy. Yet mother and fetus do not share one identical bloodstream. The child develops blood according to its own inherited instructions. If the mother is Rhd negative and the fetus inherits the D antigen from an Rhd positive father, the red blood cells bear a difference invisible to feeling, invisible to sight, and, before modern medicine, invisible to every person waiting for the birth. For much of pregnancy, this difference may cause no harm. The placenta is not simply an open channel through which maternal and fetal blood pour together. It is a specialized interface, maintaining separation while enabling exchange. Small crossings of fetal cells into the mother's circulation can occur, however, and the likelihood may increase during delivery, when the placenta separates from the uterine wall and the physical work of birth can bring blood into closer contact. Such fetal-maternal hemorrhage may also occur during miscarriage, ectopic pregnancy, abdominal injury, bleeding in pregnancy, invasive procedures, or attempts to turn a baby before delivery. Often the quantity is extremely small, almost absurdly small when compared with the consequence. A few fetal red cells carrying the D antigen may enter the circulation of a mother whose immune system has never encountered that marker on her own cells. To her immune defenses, They do not announce themselves as beloved, expected, or half inherited from a partner. They are red cells displaying a foreign antigen. The immune system has no understanding of kinship. That is one of the most unsettling truths in this story. Its power depends on discrimination self from non-self, familiar tissue from unfamiliar signal, harmless pattern from threat. In most of daily life, this ability protects the body with astonishing precision. It recognizes viruses, bacteria, parasites, and altered cells. It forms memories of dangers encountered before. It responds more quickly and forcefully when those dangers return. Yet the same machinery can act in circumstances where no infection exists. In the case of an Rhd negative mother exposed to enough Rhd positive fetal red cells, her immune system may begin a process called alloimmunization, or sensitization. It identifies the D antigen as something not belonging to her own red cells and develops anti-D antibodies directed against it. Nothing in that reaction is evidence that the fetus is unnatural. It is evidence that immunity is a system of molecular recognition, not of human meaning. Sensitization does not always happen after an Rhd incompatible pregnancy, even without prevention. The amount and timing of fetal blood transfer vary. The mother's immune response varies. BO differences between mother and fetus can sometimes reduce the chance of Rh sensitization because incompatible fetal cells may be cleared rapidly before a strong anti-D response develops. Biology does not follow a single rigid script. This unevenness once contributed greatly to confusion. One Rhd negative mother might deliver several Rhd positive children without an obvious crisis, while another might become sensitized after a birth or pregnancy loss and face danger in later pregnancies. Even today, doctors assess risk through blood tests rather than assuming that every incompatible pairing produces the same outcome. What is known is that once clinically significant anti-D antibodies form, they can persist as immune memory, ready to matter profoundly if another D-positive fetus begins to grow. At first, the mother may remain entirely unharmed. There is no fever announcing that her immune system has learned the antigen, no pain marking the moment her future pregnancies have changed, no outward wound. If the first exposure occurs late in a first delivery, that infant may be born safely before maternal antibodies reach levels capable of severe destruction. The family may leave the birth room with every reason to believe future pregnancies will follow the same path. But in a later pregnancy, if the fetus is again Rhd positive, the mother's immune response no longer begins from ignorance. Her anti-D antibodies are of a class capable of crossing the placenta. They pass from maternal circulation toward the fetus, a route normally used beneficially to provide the unborn child with maternal immune protection. This time, among the antibodies crossing that boundary are those trained to recognize the D antigen. They bind to the fetus's red blood cells and mark them for removal and destruction. A fetus depends on red blood cells for the same fundamental reason every human body does: they carry oxygen. As antibody-coated cells are cleared, the fetal body attempts to replace them at greater speed. The bone marrow increases production. When the demand becomes extreme, other organs, especially the liver and spleen, are pulled into emergency blood production. Immature red cells may spill into circulation before they are fully ready, which gave rise to the older name erythroblastosis fetalis. In moderate disease, the child may be born anemic and later become severely jaundiced as broken-down red cells release bilirubin. In severe disease, anemia becomes life-threatening before birth. The heart must pump harder to deliver oxygen through a depleted bloodstream. Fluid can collect in the tissues and body cavities. The placenta may enlarge. The fetus may develop hydrops fetalis, swollen and failing within the womb. Without intervention, death may come before labour ever begins. This is a kind of catastrophe without noise. There is no battlefield, no visible predator, no dramatic collapse of walls or cities. A child can be endangered inside a mother who appears physically well, while the force causing harm consists of antibodies so small they remain beyond any unaided eye. For families before effective diagnosis and treatment, the pattern might unfold with particular cruelty. An early child thrives, another is lost late in pregnancy. A newborn is born yellow and dies soon after. Later pregnancies may fare worse as antibody levels increase, or as the fetus inherits the antigen again. Not every child is affected, because the father may carry only one D-associated copy and pass either Rhd positive or Rhd negative status to different children. One infant may escape because it is Rhd negative like the mother, while a sibling is attacked because it inherits D, without blood typing. Even the apparent randomness is unbearable. Life and death alternating within one household according to an inherited difference no one understands. Long before the mechanism was discovered, severe newborn jaundice must have been one of the outward signs most likely to be recognised. A slight yellowing in newborns is common and often manageable, but in hemolytic disease, bilirubin can accumulate quickly and dangerously after red cells are destroyed. At high concentrations, bilirubin can cross into the brain and cause permanent neurological injury, a condition historically associated with kernicterus. An infant who survives the initial anemia may still be left with devastating damage. Another lesser-known aspect of the condition is that the fetus may be profoundly anemic before bilirubin appears dramatic after birth, because during pregnancy the placenta helps remove bilirubin through the mother's circulation. A baby can therefore be in grave danger in the womb without displaying the postnatal yellow colouring that later makes the destruction visible. The disease changes its face at birth. Before delivery, anemia and heart failure dominate. Afterward, jaundice and bilirubin injury become major threats. Once physicians recognise that maternal antibodies are responsible, pregnancy becomes a scene not merely of waiting but of surveillance. Blood from an Rhd negative pregnant woman can be tested for unexpected antibodies. If anti-D is absent, the focus is prevention. If it is already present, the pregnancy may need careful monitoring to determine whether the fetus is at risk and how severe the anemia might become. In modern practice, this can involve paternal or fetal antigen assessment where appropriate, measurement of antibody levels, and ultrasound techniques that estimate fetal anemia by observing the speed of blood flow through a vessel in the fetal brain. A severely anemic fetus can sometimes receive blood by intrauterine transfusion, an intervention so precise and improbable that it would have seemed like sorcery to the families who once endured repeated losses without explanation. A needle guided into fetal circulation can deliver compatible red cells before birth, buying time until the child is mature enough to be delivered and treated outside the womb. Yet the existence of such interventions should not blur the original severity of the condition. Intrauterine transfusion is not a casual remedy. It is specialist care for a pregnancy already endangered by antibodies. Even modern management requires expertise, monitoring, compatible blood supplies, and access to medical systems capable of sustaining both mother and fetus through a high-risk course. The great achievement in Rh disease is not only the ability to rescue some affected children, it is the ability to prevent many mothers from becoming sensitized in the first place. Before that preventive step became possible, diagnosis offered knowledge but could not erase the risk already written into immune memory. There is an emotional temptation to interpret the mother's antibodies as her body rejecting her child. Popular accounts sometimes phrase the condition in terms almost designed to sound ominous, as though Rh-negative blood refuses the blood of ordinary humans. That framing is both inaccurate and unnecessarily cruel. The mother does not will the response, and her body is not judging the child as alien. Maternal-fetal immune conflict occurs in several settings involving antigens beyond Rhd, and Rhd itself is only one particular marker capable of provoking this reaction. The immune system is responding to cells that differ from its own, much as it may respond to transfused blood. The tragedy comes from the placenta's extraordinary connection. Antibodies built in one body can reach red cells in another body still dependent upon it. The same biological closeness that makes gestation possible also creates the pathway along which harm may pass. The claim that Rh incompatibility proves two ancient human lineages were never meant to mix collapses when placed beside the actual genetics. Rhd positive and Rhd negative partners conceive healthy children constantly. Today, with proper preventive care, many Rhd negative mothers carry Rhd positive babies safely, without ever forming anti-D antibodies. Even before prevention, many incompatible pregnancies do not lead to sensitization or severe disease. The problem is not that two categories of people are biologically forbidden from joining, it is that an inherited red cell antigen can provoke an immune response in specific circumstances. Similar principles shape organ transplantation and blood transfusion. They do not imply that compatible donors and recipients belong to separate forms of humanity. The mystery is not a barrier imposed by ancient blood. It is a vulnerability created by the remarkable sensitivity of the immune system. Still, the old grief should not be lost beneath technical explanation. For a mother in an era before routine blood grouping, before antibody screening, before anti-D injections, the pattern would have seemed as though the future itself had turned hostile. She might remember the living firstborn and then watch subsequent pregnancies shadowed by fear. No one around her could tell her why her healthy body could not protect the child within it. In some communities, repeated infant loss might bring stigma, superstition, or blame. The father's inherited contribution would remain invisible. The mother, already grieving, might carry responsibility in the eyes of others for a process over which she had no control. Medical language arrived late to such rooms. Prevention arrived later still. Their negative story is therefore grounded not in secret power, but in an unbearable intimacy. Bloodlines do matter, not as mystical rank, but as inheritance. A father can pass an antigen to a child. A mother can lack it. A crossing of cells can educate her immune system, and a later infant can pay the cost. In the modern laboratory, the sequence can be diagrammed neatly. In lived history, it was not neat at all. It was a mystery repeated from cradle to cradle. An invisible toll accumulating through generations, long before medicine found a way to stop immune memory from turning a mother's next pregnancy into danger. The dead do not arrange themselves neatly for later explanation. A parish register may contain a baptism followed almost immediately by a burial. A family Bible may record a daughter who lived for 2 days, a son described only as stillborn, another infant whose name is written and then never mentioned again. A hospital ledger from the 19th century may note jaundice, weakness, convulsions, dropsy, or a child born after death in the womb. The pages preserve consequences, not causes. No one took a vial of maternal blood, looked for anti-D antibodies, typed the father, or tested the cells of the infant. The evidence remains incomplete, and it is impossible to move backward through every old sorrow and declare which losses resulted from Rh incompatibility. Infection killed newborns, prematurity killed them, birth trauma, congenital disorders, malnutrition, unsafe delivery, and countless other hazards took children in numbers now difficult to imagine. Yet threaded through that larger field of grief, almost certainly, was the repeating pattern of Rh disease first pregnancies spared or less severely affected, later infants born jaundiced, anemic, swollen, or dead, while families watched an invisible danger return. In earlier centuries, the loss of an infant was tragically familiar, but familiarity did not make it bearable. It altered the way grief was recorded. When mortality is high, records become terse not because parents feel less, but because death has few medical details to offer and life must continue amid repeated blows. A midwife may remember that one mother's babies became yellow soon after birth, or that several died despite appearing fully formed. But memories pass with the people who hold them. Wealthy households might preserve letters describing a child who weakened strangely in the nursery. Poor families may leave little more than a name and a burial date. Even royal or aristocratic genealogies, so often mined for claims about rare bloodlines, usually cannot establish the blood groups of the mothers and infants involved. A pattern of lost heirs may invite speculation, but without testing, it remains speculation. This absence of proof has been used by some to make the past sound deliberately concealed. In truth, most of the silence comes from a simpler source before the discovery of blood groups and antibodies. There was nothing precise for records to say. The outward signs of severe hemolytic disease were visible, though they were not unique. An infant with intense jaundice might be described as deeply yellow or saffron-coloured. A severely anemic newborn could appear ghostly pale, barely able to nurse, breathing rapidly, slipping away despite warmth and care. In the worst fetal cases, a child might be delivered with extreme swelling, fluid gathering beneath the skin and within the body. A sight that physicians eventually described with clinical names, but that must have been terrifying in any century. For a long time, such conditions were classified by appearance. Hydrops fetalis meant a swollen fetus, or newborn, not one particular cause. Grave newborn jaundice could be observed without revealing why it occurred. Erythroblastosis fetalis came closer to the process by identifying the flood of immature red cells. But even that described the body's desperate response rather than the original attack. Medicine could name the ruin before it knew who had set it in motion. The sequence from one pregnancy to another was the most revealing clue and also the easiest to miss. Many families were large. Many children were lost for unrelated reasons. Pregnancies might occur with different fathers. Miscarriages could be hidden or unrecorded. An affected fetus might be Rh-D negative and safe despite an already sensitized mother, while the next inherited D and became critically ill. Nothing required the losses to arrive in a simple row. A first pregnancy could sensitize a mother during miscarriage or traumatic delivery. A transfusion given years earlier, perhaps after bleeding or surgery, could sensitize her before she ever became pregnant. An infant could survive mild disease and leave no unmistakable mark in a record. Another might die of infection after being weakened by anemia, obscuring the original cause. The true pattern was biological, but its historical footprints were scattered and easily overrun. By the late 19th and early 20th centuries, physicians increasingly recognized particular forms of newborn catastrophe as related phenomena. Some noticed that severe jaundice could run through siblings. Some recognized that fetal swelling and newborn anemia belonged to a spectrum rather than to wholly separate illnesses. Autopsies and microscope work showed immature red cells filling blood-making organs. The liver and spleen of affected infants could be enlarged from their frantic effort to compensate for destruction in the bloodstream. Yet even skilled clinicians faced a missing link. Why did a healthy mother produce these sick infants? Why did the disease often intensify in later births? The mother did not appear infected, poisoned, or deformed. Her body could deliver a perfectly healthy child before it appeared to fail subsequent ones. In the absence of an immune explanation, the story could be interpreted in ways that deepened a woman's misery: weakness of constitution, maternal fault, inherited degeneration, or simple fate. There is a peculiar cruelty in diseases whose cause is not only hidden, but mistakenly assigned to the person already carrying the grief. Pregnancy and birth have always attracted judgment. When a baby died, a woman could be blamed for work she had done, foods she had eaten, emotions she had felt, prayers she had neglected, or qualities believed to reside in her body. Heart disease gave no sign by which she could defend herself. A sensitized mother did not feel the antibodies moving across the placenta. She could follow every available instruction and still lose an Rh-D-positive baby. No remedy offered by family tradition could remove an immune memory once formed. Her next pregnancy may have been marked from the beginning by the recollection of earlier loss, while everyone around her waited without knowing that the danger had a measurable cause. Beneath later stories of special blood and secret powers lies this older reality. Rh-negative status did not confer mastery. In unprotected pregnancies, it could expose families to a pattern of pain they had no means to explain. The emergence of modern hospital medicine begins to make this toll more legible. As births increasingly come under medical observation in some countries, clinicians record fetal deaths, profound jaundice, anemia, and high drops with greater regularity. Laboratory work begins to compare mothers, fathers, and infants. Once Levine and Stetson identify the maternal antibody connected to a stillborn infant and a transfusion reaction, older observations rearrange themselves into a new picture. The mother is not generally ill, she has become immunized. The first contact with incompatible fetal red cells has prepared her blood to respond more powerfully later. The disease that seemed to strike one infant after another without reason is an immune event governed by inheritance and exposure. For some women, this knowledge must have landed with almost unbearable force. The dead children had not vanished through vague misfortune. There had been a cause. Science had found it only after they were gone. Once the cause is recognized, doctors face a new moral pressure. To diagnose a danger without being able to prevent it means watching more closely, while still knowing that observation alone may not save a child. In the middle decades of the 20th century, management of affected infants improves through exchange transfusion. In a severe case after delivery, clinicians can slowly remove portions of an infant's antibody-laden, bilirubin-producing blood and replace it with compatible donor red cells. It is painstaking work. The newborn may be tiny, weak, and already threatened by brain injury or heart failure. Blood must be chosen carefully. The procedure can reduce bilirubin, remove circulating maternal antibodies, and restore functioning red cells, saving children who might otherwise die. But every such rescue begins after the mother has been sensitized and the infant has already entered danger. Even more remarkable is the attempt to treat a child before birth. In the early 1960s, physicians began carrying out intrauterine transfusions for fetuses endangered by severe hemolytic disease. At first, blood is introduced into the fetal abdominal cavity, where it can be absorbed into circulation. Later refinements allow direct transfusion into fetal vessels under imaging guidance. The image belongs almost to science fiction when set against the centuries behind it. A fetus once doomed by an invisible reaction can receive donor red cells while still inside the womb. Some babies survive because of this intervention, some do not. The procedure is risky, the pregnancies are already gravely threatened, and the specialist expertise required is not universally available. But it demonstrates something crucial. Rh disease is not destiny written by an incompatible bloodline. When its mechanism is understood, its effects can be interrupted. Before prevention becomes routine, the number of affected families is substantial. In communities with appreciable Rhd negative frequency and limited testing, a sequence of sensitization and later disease is not an exotic event. Obstetric wards see women whose antibodies threaten fetus after fetus. Pediatric services treat yellow, anemic newborns. Blood banks prepare compatible units for exchange transfusions. The disease becomes one of the major recognized causes of fetal and neonatal illness, linked to blood group incompatibility. It also creates a form of medical memory within families. An older relative may recall babies lost before a treatment became available. A mother may know that one child survived because doctors acted rapidly after birth, while another pregnancy ended with disease too severe to overcome. By the time later, generations hear the phrase Rh negative. It may already carry an inherited emotional charge, not because of an ancient secret, but because the family once paid the price of a preventable immune response before prevention existed. The toll is also unequal. Medical advances do not arrive everywhere at once, and they do not reach every patient in the same way. Blood typing, antibody screening, exchange transfusion, specialist monitoring, and compatible donor supplies depend on systems-trained staff, laboratories, transportation, records, resources, and trust in healthcare. A disease largely controlled in one country can continue harming babies elsewhere, even within wealthy societies. Gaps in prenatal care or failures to provide prophylaxis at the right moment can leave a patient sensitized for life. The past does not close cleanly when a discovery is made. It recedes unevenly, leaving pockets of old risk among people least able to avoid it. This uneven retreat helps explain why the phrase conspiracy of silence can find listeners. Some people learn about Rh-negative blood only after a difficult pregnancy, a lost child, or a frightening medical warning. They may feel that something so consequential should have been discussed far earlier. Others encounter family stories of infant death for which no explanation was offered at the time, where medicine has communicated poorly, where grief has been dismissed, or where access to care has failed. Distrust can take root, but the silence before the 20th century was not evidence that authorities hid knowledge of a separate bloodline. There was no knowledge to hide, and the silence that persists in individual families often reflects sorrow, inequality, medical complexity, or the simple difficulty of speaking about children who died before they could be known. By the time our disease is understood, humanity can finally look backward and recognize at least part of the shadow it cast. It lies in unnamed infants and interrupted genealogies, in mothers pregnant again beneath the memory of small graves, in physicians who saw the pattern before they had the key, and in the first newborns rescued by carefully exchanged blood. A rare inherited difference has left a real historical legacy, but not the legacy promoted by legends of hidden abilities or forbidden origins. Its legacy is written in absence children who might have lived had their mothers been born in an age with one more piece of knowledge. Then, almost abruptly on the scale of history, that knowledge takes another step. Medicine finds a way not merely to treat the attacked child, but to prevent the maternal attack from beginning. The invisible toll, endured for uncounted generations, is about to meet the injection that changes its course. The solution, when it finally comes, seems almost paradoxical. A mother's immune system has become dangerous to an Rh-positive fetus because it has learned to recognize the D antigen. The way to prevent that learning is not to suppress pregnancy, not to change the child's bloodline, and not to alter the mother's genes. It is to give the mother antibodies against the very antigen that might otherwise provoke her own immune response. These donated antibodies, contained in anti-D immune globulin, move quickly to clear or conceal any Rhd-positive fetal red cells that enter her circulation before her immune system mounts a lasting response of its own. The mother is protected from forming the memory that could threaten a later child. It is an elegant act of biological misdirection, a temporary shield placed over a moment of exposure, preventing the immune system from turning an inherited difference into a permanent hazard. By the mid-20th century, researchers understand enough about hemolytic disease to see the shape of the problem clearly. Rhd-negative women who have not yet developed anti-D antibodies are not inevitably destined to lose children. The danger begins with sensitization. Prevent that first immune awakening, and later, Rh-D-positive pregnancies may proceed without the attack that once devastated families. Several researchers and clinical teams pursue this possibility, working from the recognition that antibodies already present in a mother's blood can affect what happens to foreign red cells. The scientific route is not entirely intuitive. Antibodies are usually imagined as the cause of disease and Rh incompatibility. Here, carefully supplied antibodies become the means of prevention. They remove the triggering cells so efficiently, or alter their presentation to the maternal immune system so successfully, that the mother does not develop her own durable anti-D response. The idea is tested first in settings where exposure can be controlled and followed. Rh-D negative volunteers exposed to Rh-D positive red cells are given anti-D preparations, and researchers watch to see whether their immune systems later become sensitized. Clinical trials involving Rh-D negative mothers then show that anti-D administered after the birth of an Rh-D positive child dramatically reduces the likelihood of future alloimmunization. The result is one of the great quiet revolutions in reproductive medicine. A process that has haunted human birth for as long as the relevant blood variants have existed can now be interrupted with an injection given at the correct time. It does not erase sensitization already established. It does not treat every form of fetal blood group disease, but for the leading cause of severe Rh-associated hemolytic disease, it changes the future. The first widespread strategy focuses on the period after delivery. Birth is one of the most important moments for fetal red cells to pass into maternal circulation, particularly when the placenta detaches. If an Rh-D negative mother gives birth to an Rh-D positive infant and has not already become sensitized, anti-D can be given soon afterward, before her immune system forms its own enduring antibody response. The infant's blood is typed, the mother's status is known, a treatment follows, often within days of delivery, and a later pregnancy that might once have been endangered is protected before it even exists. To a patient, it may look like a brief clinical routine after the overwhelming experience of birth. To history, it is the closing of a door through which grief had entered families for generations. Later, prevention becomes even stronger when anti-D prophylaxis is also offered during pregnancy, commonly in the third trimester, because small fetal maternal blood transfers can occur before labour. Additional doses may be required after events that increase the chance of fetal blood entering maternal circulation miscarriage, or termination in circumstances where prophylaxis is indicated under local guidance. Bleeding during pregnancy, abdominal trauma, invasive procedures such as amniocentesis, external turning of a breech baby, or other sensitizing events. Medical practice varies in timing, dosing, thresholds, and testing protocols among countries and over time, but the principle is consistent. An Rh-D negative pregnant patient who is not already sensitized can often be protected from developing anti-D antibodies when exposure risk is recognized and anti-D immune globulin is provided appropriately. This protection depends on a distinction easy to overlook. Anti-D prophylaxis prevents sensitization. It cannot undo it. If a woman has already developed her own immune anti-D antibodies, an injection of prophylactic anti-D does not erase that memory. Her future pregnancies with an Rh-D positive fetus may still require careful monitoring and, if disease becomes severe, fetal treatment or early delivery. This is why antenatal screening matters so much, early in pregnancy. Blood tests establish the mother's ABO and Rh-D status and look for red cell antibodies. If she is Rh-D negative but has no anti-D antibodies, she may be eligible for protection. If she already carries clinically significant antibodies, the question shifts from prevention to risk assessment. The blood test is not simply paperwork performed at the beginning of prenatal care. It is a fork in the road, identifying whether a danger can still be prevented or must now be watched. For the first generation of women to benefit widely from prophylaxis, the change could be astonishing. A mother may have grown up hearing of relatives who lost babies after apparently ordinary pregnancies. She may have seen older women in her family carry a fearful respect for the minus sign in their blood type. Then she enters a maternity clinic and receives an injection that sharply reduces the threat to children she may have years later. The sorrow of previous generations does not disappear. The children already lost are not returned, but the pattern is no longer required to repeat itself. A medical intervention, derived from painstaking laboratory work rather than ancestral revelation, breaks a cycle that once seemed like fate. The success becomes measurable. As anti-D prophylaxis is adopted in well-resourced healthcare systems, the number of babies severely harmed or killed by Rh-D alloimmunization falls dramatically. Exchange transfusions and intensive neonatal treatment remain necessary for cases in which sensitization has occurred or disease results from other antibodies, but the former stream of Rh-D disease begins to shrink. Obstetric medicine experiences a rare form of victory. Not merely diagnosing a terrible condition earlier, not merely making its consequences slightly less severe, but preventing large numbers of cases before the fetal bloodstream is ever attacked. Because the reduction occurs over years rather than in one spectacular event, it can be overlooked in public memory. Diseases controlled by prevention often become invisible precisely because prevention works. The children who would once have grown ill are simply born healthy. The danger erased so early that no family story forms around it. That invisibility creates a strange reversal. When Rh disease was common, its effects were obvious, but its cause was mysterious. Once its cause is understood and prevented, the mechanism is clear to medicine, but its former devastation becomes less visible to the public. People may hear that an Rh-negative mother receives a shot during pregnancy and assume the matter is minor, bureaucratic, perhaps unnecessary. Others encounter online claims that the injection exists because Rh-negative women have unusual blood requiring control, or because some larger secret is being suppressed. The truth is more grounded and more impressive. The injection is not evidence of a hidden division within humanity. It is evidence that medicine learned to intervene at the precise point where immune memory would otherwise arise. Anti-D immune globulin is itself tied to blood donation and to the quiet participation of people whose antibodies can be used to protect strangers. Preparations are derived from human plasma containing anti-D antibodies, collected, purified, processed, and controlled according to medical standards. This dependence on donors adds another human layer to the story. One person's carefully screened antibodies, which in an unplanned pregnancy context could represent danger, can be transformed into a preventive medicine that helps another woman carry future children safely. Blood here is neither symbol nor secret code. It is a shared biological resource, managed through trust, testing, and the often invisible labour of donation systems. The history of Rh-negative pregnancy becomes not only one of molecular conflict, but of people protecting infants they will never meet. The prevention is extraordinarily effective, but it is not universal magic. Failures can occur when prophylaxis is not given, when exposure is larger than expected and dosing is insufficient without proper assessment, when care is interrupted, or when sensitization began before treatment. Some women develop antibodies to other red cell antigens, such as KEL or certain Rh system antigens besides D, for which routine prevention does not provide the same solution. A fetus can suffer serious hemolytic disease even when the famous D incompatibility is not the culprit. The medical story therefore does not end with a single injection. It expands into antibody screening, specialized blood banking, fetal surveillance, neonatal care, and the need for rare compatible donors. No single explanation fully accounts for every case of maternal-fetal blood incompatibility, and no prevention program reaches its full promise where healthcare access is weak. That inequality is where the triumph remains unfinished. In regions with reliable prenatal care, testing, blood supplies, and prophylaxis programs, severe disease from maternal anti-D can become uncommon. In places where pregnant patients are not typed early, where anti-D is unavailable or unaffordable, where births occur far from medical facilities, or where records fail to follow a woman through pregnancy and delivery, an ancient hazard can remain dangerously present. The molecule has not changed. The difference lies in whether knowledge is accompanied by access. A child's chance of escaping Rh disease may depend not only on inherited blood type, but on whether a health system can provide a vial of preventive medicine at the right moment. That is not a conspiracy of silence, but it is a moral failure whenever a known, preventable danger persists through neglect or scarcity. It is also why fear around Rh-negative blood can never be answered only with dismissal. People who ask why their blood matters may be responding to a genuine medical warning, a complicated pregnancy, or a family history no one explained clearly. The right answer is not to replace one mystery with another. Rh-negative status matters because pregnancy can expose an Rhd-negative person to fetal Rhd-positive cells, causing antibodies that endanger future Rhd-positive fetuses. The prevention is a carefully timed medical intervention that blocks that sensitization in most eligible cases. Nothing in this mechanism supports alien ancestry, hidden powers, or a forbidden union between different kinds of people. Yet nothing about it is trivial. It is one of the clearest examples of modern medicine reaching backward into a recurring human tragedy and stopping it from moving forward. For a mother who receives anti-D today, the moment may pass quickly a needle, a note in the medical record, a recommendation to return for appropriate follow-up, another step in a pregnancy already filled with appointments. She may never see the danger it prevented. Her next child may be born with the D antigen and remain entirely well, not because the inherited mismatch vanished, but because her immune system was prevented from remembering it as an enemy. The silent difference in their blood remains. What changes is its power to harm. After centuries in which the minus sign could cast a shadow over birth without anyone knowing why, the shadow finally recedes. Rh-negative blood has not been cured because it is not a disease. An Rhd-positive fetus has not been altered because it does not need alteration. Instead, medicine has learned to stand at the narrow point where two inherited realities meet and prevent that meeting from becoming war. The supposed hidden power of this bloodline is not found in myth, superiority, or secret history. It is found in the accumulated human intelligence that recognized an invisible danger, followed it through loss and uncertainty, and placed into a mother's hand the possibility that her children would no longer inherit the tragedy. Once anti-D prophylaxis begins to prevent many of the most devastating pregnancy outcomes, Rh-negative blood does not lose its significance. It simply becomes significant in another place, often far from the intimate world of the maternity ward. In emergency rooms, operating theatres, ambulances, military hospitals, disaster sites, and blood banks illuminated long after midnight, the minus sign remains a practical matter of life and death. A patient bleeding rapidly after a collision does not arrive with time for a long explanation of ancestry. A woman hemorrhaging during delivery may require blood before anyone knows whether the danger will deepen. A child undergoing surgery, a victim of violence, a patient whose red cells are being destroyed by illness or treatment may all depend on blood that can enter circulation without provoking a new crisis. In those moments, rare blood is not an emblem of secret identity. It is a scarce medical resource, one that must be found, tested, transported, and given before a body runs out of the oxygen-carrying cells, it needs to remain alive. Blood transfusion depends on compatibility, but compatibility is never quite as simple as the public shorthand implies. Most people know the broad outline. O-negative red cells are often called the universal emergency donor type because they do not display A, B, or D antigens, the three markers most immediately important when blood must be given before a recipient's full type is confirmed. In a major emergency, O-negative units may be placed into a body whose identity is still unknown, buying time while laboratories perform testing and clinicians stop the source of bleeding. That label, universal, has a powerful sound, and it has helped surround Rh-negative blood with an aura of unusual purity or superiority. But even O-negative blood is not truly invisible to every immune system. Red cells carry many antigens beyond A, B, and D. A recipient with antibodies against another clinically important antigen may still need specifically matched blood. O-negative is extraordinarily useful in emergencies, especially when avoiding D exposure is important. But it is not magical blood that can move without consequence into every person under every circumstance. Its usefulness creates vulnerability. O-negative donors make up a minority of most populations, while their blood is used disproportionately in emergency release, neonatal care, certain transfusions for patients who must avoid sensitization, and situations in which typing cannot safely delay treatment. Blood banks therefore manage a constant tension, preserve O-negative supplies for those who need them most urgently, yet release them quickly enough when hesitation could cost a life. When shortages occur, the problem is not theoretical. A unit of compatible blood cannot be manufactured instantly in response to an accident. It must already have been donated by another human being, screened for infectious risk, processed, stored under controlled conditions, inventoried, and delivered to the place where it is needed. A person's rare or uncommon type becomes valuable because it can answer another person's emergency, not because it grants its carrier hidden strength. The RhD issue is especially important in transfusions because exposure can sensitize an RhD negative patient, just as fetal blood can sensitize an RhD negative mother. If an RhD negative person receives RhD positive red cells, their immune system may form anti-D antibodies. This is not equally consequential for every patient in every circumstance. In life-threatening hemorrhage, doctors sometimes must balance the immediate danger of death against later sensitization risk, particularly when supplies are limited. But for an RhD negative girl or woman who may later carry an RhD positive fetus, preventing anti-D formation can protect future pregnancies. The unit of blood given after trauma today can matter years later, in a child not yet conceived. This interweaving of emergency medicine and reproductive future is one of the quieter reasons blood banks treat Rh status with such care. For many Rh-negative patients, finding compatible red cells is manageable because the common D-negative forms are well represented in donor programs, serving populations where the trait is relatively frequent. Difficulty increases when a patient has formed antibodies beyond anti-D, especially after multiple transfusions or pregnancies. Each exposure to foreign red cell antigens can encourage the immune system to recognize additional differences. A person receiving regular transfusions for chronic disease may gradually become difficult to match, because safe blood must avoid several antigens at once. This is why blood banking is less like sorting bottles into 8 familiar categories, and more like managing a vast biological library, in which some readers require a book printed in an unusually specific edition. The letters and plus or minus sign on a donor card are only the front cover. The need becomes still more urgent in ethnically diverse societies. Red cell antigen frequencies vary among populations, shaped by the same ancestry and population history that made the RhD map so compelling. A patient is often more likely to find closely matched uncommon antigen combinations among donors with related ancestral backgrounds, though no person or community can be reduced to ancestry alone. When a national donor pool fails to represent the people who may need transfusions, patients with rare combinations or multiple antibodies can face longer, more difficult searches. A modern blood system therefore depends not only on medical technology, but on broad participation and trust. The story of rare blood is not a story of exclusive status. It is a story of interdependence. The more varied the donor community, the better the chance that a stranger's exact need can be met. Some blood requirements are so unusual that routine local inventories are insufficient. Rare donor registries connect blood services across regions and countries, identifying people whose antigen profiles may be crucial to patients with difficult antibodies. Frozen red blood units can sometimes be stored for extended periods, held against the day when a matching patient appears. The people who provide these donations may never know whose life their cells enter. Their blood may cross borders packed in validated cold chain systems, accompanied by laboratory records and urgent communications rather than ceremony. In legends, rare blood is guarded by secret orders. In reality, it is guarded by technicians checking labels, donors returning for appointments, couriers moving coolers through airports, and specialists comparing antigen profiles while a clinical team manages a patient whose options are narrowing. At the far edge of this world lies a phenotype so rare that it has become famous under a misleadingly glamorous nickname golden blood. This is Rh null, a condition in which red blood cells lack all antigens of the Rh system. Not merely the D antigen absent in ordinary Rhd negative blood, the difference is immense. A typical Rhd negative person does not express D, but still carries other Rh antigens such as combinations related to C, E, or E. An Rh null person lacks the Rh antigen complex across the red cell surface. Only a tiny number of such individuals have been identified worldwide, and their blood can be extraordinarily important for certain patients who have antibodies reacting against common Rh antigens. Because Rh null cells lack those targets, they may serve as uniquely compatible donor blood in exceptionally rare situations. This medical value has encouraged the nickname, as though the blood itself were treasure. Yet Rh null is not a biological upgrade. The Rh complex plays a role in the stability of the red cell membrane, and people with Rh null blood can experience fragile or unusually shaped red cells, ongoing low-grade destruction of those cells, and chronic anemia of varying severity. The same absence that makes their donated cells precious to a particular recipient can make their own medical care difficult, if an Rh null individual ever requires transfusion. Finding compatible blood can be extraordinarily challenging. They may rely upon other extremely rare donors or stored units of their own blood where circumstances allow. The golden image disguises the truth rarity can bring responsibility and risk, not superiority. A person with Rh null blood may be valuable to medicine in a very narrow way, while also living with a vulnerability ordinary Rh negative people do not share. Popular accounts often blur this distinction. They begin with Rh negative blood, speak of its relative rarity, then slide into stories about golden blood, implying that all negative individuals share some extreme, mysterious condition. They do not. Millions of Rh negative people carry ordinary, healthy red blood cells lacking the D antigen. Rh null is a separate and extraordinarily uncommon phenotype, involving the absence of all Rh antigens. Confusing them is like treating every person with an uncommon eye colour as though they possess a unique genetic syndrome. The mistake is attractive because it amplifies the mystery of one rare blood phenotype is almost vanishingly scarce and medically precious. Then perhaps every negative blood type hides the same importance. But transfusion science requires precision precisely where myth thrives on vagueness. There are other unusual Rh cases as well, including partial and weak expressions of D, and combinations that may complicate whether a person should be treated as positive or negative in particular medical circumstances. Someone with a partial D antigen may produce antibodies against the portions of D they lack if exposed to conventional D-positive cells. Molecular typing can therefore matter when routine tests do not fully reveal transfusion or pregnancy risk. These variants show once again that Rh is not a binary division between two ancient camps. It is a complex blood group system, generated by related genes and rearrangements, expressed differently across individuals and populations. The deeper medicine looks, the less plausible any simple hidden bloodline story becomes. The shortage problem also exposes the emptiness of claims about power. A person with O-negative blood may be urgently encouraged to donate, particularly if supplies are low. That request can be flattering, even identity-forming. Donors may take pride in knowing their blood is especially useful in emergencies. Such pride is deserved when it leads to generosity, but medical importance is not personal supremacy. The blood is valuable because it avoids antigens that could harm recipients before detailed matching is complete. The donor is still susceptible to disease, injury, aging, and all the ordinary limits of a human body. If that same person develops bleeding and requires antibodies to other blood antigens, they may themselves become dependent on a system of donors and specialists. There is no hierarchy here, only compatibility. In fact, blood banking quietly reverses the mythology of separation. Myths of rare blood suggest a group apart, biologically removed from others by ancient origin or special qualities. Transfusion medicine reveals the opposite. The meaning of rare blood emerges through connection. O-negative cells matter because they can sustain unknown patients in urgent need. Rh null cells matter because they may fit a recipient almost no other donor can safely reach. Antibody screening matters because a pregnancy joins two inheritances within one body. Rare donor registries matter because someone on one side of the world may be the only safe match for a stranger on another. Blood is intimate ancestry, but it is also shared survival. There is gravity in a refrigerator drawer containing rare frozen units. The bags do not glow. No secret is visible inside them. They hold dark red cells suspended and labelled with the care required for a resource that may not be replaceable on demand. Somewhere, a patient may already be bleeding or awaiting surgery, or facing a transfusion reaction because the usual stock cannot be used. Somewhere else, a donor carries an antigen pattern that happens to answer that need. Between them stands a chain of science and civic trust, built from the understanding that the invisible differences in human blood are not reasons to divide people into superior and inferior lines. They are reasons to know one another more carefully, to prepare, and sometimes to give. The Rh-negative mystery began in the destruction of fetal blood by an antibody no one yet understood. It now opens outward into a global network created because blood cannot simply be replaced by any blood at all. The more closely medicine studies these variations, the more intricate the human picture becomes. Rare blood can save. Rare blood can place its carrier at risk. Rare blood can require extraordinary matching. But it does not unlock a hidden faculty, a superior perception, or an inheritance from beyond the human past. Its real power appears only in moments when another body is failing, and a compatible donation crosses the distance between strangers, carrying nothing mystical through the vein, only red cells, oxygen, and a little more time. A rare blood type does not need a legend in order to matter. It already enters the world through scenes charged with emotion, and the pregnancy watched more closely than expected. The injection given to protect a future child, the emergency request for donors, The hospital explanation that one inherited marker may decide which blood is safe, but rarity rarely remains only medical in the human imagination. Once a trait is hidden beneath the skin, inherited through families, unevenly distributed across populations, and capable of producing serious consequences when positive and negative blood meet, it begins to gather meanings far beyond the laboratory. People ask not only what it does, but what it means. They begin to wonder whether the body is preserving a secret that history forgot. And around B-negative blood, that wondering has produced one of the strangest modern mythologies attached to an ordinary human gene. The claims vary, but their pattern is remarkably consistent. R-negative people are said to have lower body temperatures, unusual sensitivity to heat or light, heightened intuition, exceptional empathy, psychic awareness, unusually vivid dreams, extra vertebrae, striking eyes, higher intelligence, or an instinctive feeling of being different from everyone around them. In more elaborate accounts, these supposed features become signs of a lost lineage, descendants of ancient gods. Survivors of Atlantis, remnants of a superior prehistoric race, offspring of non-human visitors, or carriers of a genetic alteration deliberately hidden from the public. The medical reality of pregnancy incompatibility is often pulled into the story as its dramatic proof. A mother's immune system can attack the blood cells of an Rhd-positive fetus, the argument says, because the two types were never meant to mix. A normal immunological process is transformed into the imagined border between ordinary humanity and something older, purer, or not entirely of Earth. The theory has the shape of a mystery because it begins with facts. Rhd-negative blood is less common globally than Rhd-positive blood. Its frequencies do differ among populations. It is clinically important in pregnancy and transfusion. The absence of the D antigen can matter enormously when incompatible red cells cross into a person who lacks it. All of that is true. The distortion happens when these facts are made to support claims they do not support. No credible evidence links ordinary Rhd-negative status to psychic ability, unusual intelligence, a separate species, ancient non-human ancestry, or a suppressed genetic power. No accepted genetic research finds that Rhd-negative people possess a hidden cluster of extraordinary abilities associated with the absence of the D antigen. No archaeological discovery has identified an advanced lost population distinguished by this blood trait. No medical finding shows that Rh incompatibility means positive and negative people are fundamentally unfit to reproduce together. The difference is real. The mythology built around it is unsupported. The alien bloodline version of the story often begins with a misleading premise that because Rhd-negative blood lacks a factor present in most people, it must have come from outside ordinary human evolution. But absence is not foreignness. A gene can be deleted, altered, weakly expressed, or inherited in different forms through common biological processes. Populations can carry such variations at different frequencies because of founder effects, genetic drift, migrations, and isolation. The genetics of the Rh system is complex, but it belongs entirely within the known structure of human variation. If Rhd-negative people represented the descendants of non-human interbreeding, or even of an unknown separate human species, researchers would expect far more than the absence or altered expression of one red cell antigen. The human genome does not conceal such an origin behind one hospital blood type symbol. It would leave signals across many regions of DNA. Signals that modern genetic research would be capable of examining. None supports the alien bloodline claim. There is a revealing contradiction inside many of the legends. Rhd-negative blood is sometimes described as too rare and too biologically different to have arisen in ordinary humanity, yet it is also claimed to be widespread among chosen descendants scattered through modern populations. It is portrayed as evidence of extreme separation, though Rhd-positive and Rhd-negative people have been having healthy children together throughout human history. It is treated as a sign of physical superiority, though its clearest distinctive medical consequence before prevention was an increased risk to certain pregnancies. When Rh null blood appears in the discussion, the contradiction becomes stronger still. Its extraordinary rarity and transfusion value are borrowed to make ordinary Rh-negative blood sound unique and elevated. While the genuine medical vulnerabilities associated with Rh null are usually omitted, myth takes only the fragments that glitter and leaves behind whatever returns the story to ordinary biology. The list of supposed Rh-negative characteristics operate in much the same way. Many describe experiences broad enough to fit millions of people feeling different, having strong intuition, disliking hot weather, being curious about origins, having vivid dreams, sensing emotions intensely, experiencing unexplained fatigue, or possessing features such as light eyes or reddish hair that vary for reasons unrelated to Rhd status. A person who discovers they are Rh-negative may read such a list and recognize themselves in several points. This does not require fraud or foolishness. Human beings are excellent at finding personal meaning in loosely defined descriptions, especially when those descriptions offer an explanation for a feeling of uniqueness or isolation. Once someone accepts the central idea, every matching detail appears significant while every mismatch becomes easy to ignore. A clinical label turns into a private biography. The emotional attraction becomes stronger for people who learned about their blood type through crisis. Someone who has experienced pregnancy loss, an antibody warning, a rare transfusion need, or an urgent search for compatible blood may understandably feel that their biology has placed them in a category ordinary explanations do not fully capture. Medicine can be precise without being emotionally satisfying. It may explain antigens and immune response while leaving the person alone with grief. fear, or the sense that their body has betrayed them. Into that gap comes a story that replaces vulnerability with distinction. The blood did not create danger. It reveals special ancestry. The loss was not random suffering within human biology. It was evidence of a deeper conflict. The person is not merely one patient among many. They are a bearer of something rare and hidden. Such ideas can be profoundly comforting even when they are wrong. That is why simply mocking them misses part of the story. The myths grow where mystery, identity, and pain overlap. They also grow because the genuine science is complicated. It is easier to say that Rh-negative people come from a separate ancient lineage than to explain the Rhd and Rhce genes, antigen expression, maternal sensitization, varied global frequencies, ancient admixture, population bottlenecks, and the limits of what any one genetic marker can tell us about prehistory. Myth offers a single cause for everything rarity, pregnancy risk, regional distribution, personal feelings, and historical uncertainty. Science offers many causes operating at different levels, plus an honest admission that some evolutionary details are still being studied. In a culture that often mistakes certainty for truth, the unsupported story can feel more satisfying than the accurate one. Ancient history provides a ready-made material for this mythology. Human civilizations repeatedly described gods descending from the sky, rulers claiming divine origin, heroes born from unusual unions, and sacred lineages protected by ritual or law. These traditions are rich sources for understanding the societies that created them. They are not medical records of Rh-negative ancestry. The habit of reading every myth of divine descent as literal evidence of alien genetics does violence both to history and to science. Ancient people used the language of divine parentage to explain kingship, legitimacy, cosmic order, fertility, power, and the dangerous closeness between human life and forces believed to govern it. A ruler described as descended from a god does not provide a blood sample. A relief showing a winged figure does not document genetic engineering. A legendary flood, star, serpent, or heavenly visitor cannot be assigned to Rh-negative blood by repetition alone. The same caution applies to claims involving ancient skulls, elongated heads, red-haired mummies, or royal families. Physical remains can reveal extraordinary stories when studied carefully. Disease, diet, migration, trauma, burial customs, deliberate cranial shaping, kinship, and sometimes preserved DNA. But unusual appearance does not automatically imply unusual blood type, and unusual blood type does not imply supernatural origin. In many societies, elongated skull shapes were created deliberately by binding an infant's head, a cultural practice rather than evidence of a separate species. Hair colour in preserved remains can be altered by time, burial chemistry, or natural variation. Royal genealogies can demonstrate political marriage strategies without establishing a global Rh-negative conspiracy. The past is filled with real strangeness. It does not require modern fantasies to become fascinating. One reason the alien theory survives is that it places uncertainty in the wrong category. Scientists may not know precisely when a particular common or HD deletion first appeared, or exactly which demographic forces best account for all modern frequencies. That uncertainty is then presented as though science cannot explain Rh-negative blood at all. But not knowing every step of a trait's deep evolutionary journey is very different from not knowing what the trait is. Medicine knows what determines the common Rhd positive or Rhd negative designation. Genetics knows how inherited Rhd variation produces the phenotype. Obstetrics understands how anti-D antibodies endanger a fetus, and how prophylaxis often prevents sensitization. Transfusion medicine understands why D compatibility matters. The open questions concern the fine-grained history of frequency and evolution, not whether Rh-negative people are ordinary members of the human species. There is also no need for a global cover-up to explain why the topic feels obscure. Most people do not study blood group systems until pregnancy, donation, surgery, or illness makes them relevant. Medical education focuses on what patients need to know for safety, not every evolutionary speculation that circulates online. Researchers investigating blood antigens publish in technical settings that rarely reach a general audience. When a person encounters an alarming video or an article claiming that scientists refuse to discuss Rh-negative origins, they may not realize that the subject has been discussed for decades in hematology, immunology, obstetrics, population genetics, and transfusion medicine. The knowledge is not forbidden, it is specialized. Specialized knowledge can look like secrecy from outside, especially when myths are presented in more dramatic language than evidence ever uses. The most seductive claim may be the idea of hidden abilities, because it shifts the discussion from ancestry to daily life. It invites a person to look inward and discover proof. Yet any serious attempt to show that Rhd negative status grants unusual mental or sensory capacities would require careful research, well-defined traits, large samples, control for ancestry and environment, replication, and results strong enough to survive scrutiny. Stories gathered from self-selected online groups cannot establish a biological effect. People attracted to a theory are more likely to report experiences that resemble it. Broad personality descriptions do not become genetic evidence because they are placed beside a blood type. The evidence for hidden abilities is not merely incomplete. No reliable foundation supports the claim. What does survive scrutiny is quieter but more enduring. Rh-negative blood is a variation shaped by human inheritance. Its frequencies may carry partial traces of population history. Its presence can matter greatly in pregnancy and transfusion. Its study helped medicine prevent immense suffering. Its rarity can connect donors to patients they will never know. These facts do not reduce the wonder of the subject. They locate it where it belongs. Not in a story of people secretly above or outside humanity, but in the astonishing complexity of humanity itself. A molecule missing from the surface of a red cell can influence which infant survives, which donor is called at midnight, which family learns a new word in a hospital corridor, and which ancient migration patterns remain faintly visible in living bodies. The myth promises that Rh negative people are different because they come from somewhere else. The evidence suggests something more intimate. They are different in one medically important respect because human beings have always varied, and because variation travels through generations with consequences no ancestor can foresee. That is not an inferior mystery. It is the real one, a blood type without supernatural powers, carried through ancient roads and modern lives, capable of inspiring both life-saving science and stories that reveal how urgently people want their biology to mean more than chance, inheritance, and survival. The story of secret blood does not require a secret institution to spread. It requires only a question, an unsettling medical fact, and a world in which answers travel faster when they promise that someone is hiding them. A person learns that they are Rh negative. Perhaps the discovery arrives during pregnancy, when a clinician explains the need for an injection. Perhaps it comes during blood donation, when the donor is told that their type is especially valuable. Perhaps it appears after a relative repeats a family story about infants lost long ago, or a rare blood match needed in an emergency. The person searches for an explanation and finds two kinds of language waiting. One is cautious, technical, and practical antigens, inheritance, sensitization, prophylaxis, transfusion matching. The other is immediate and personal rare bloodline, suppressed truth, ancient power, alien ancestry, the reason you have always felt different. One language asks the reader to tolerate complexity, the other tells the reader that the mystery is not only real, but about them. That is how suspicion begins to organize itself. It rarely starts with a vast plot described in detail. It starts with the sensation that an explanation has been withheld. Why did no one mention this blood difference before a pregnancy? Why is the injection necessary? Why are some blood types rarer than others? Why are particular populations discussed in relation to the trait? Why does a standard medical website say so little about origin stories that appear everywhere in videos and forums? A reasonable gap in understanding becomes evidence of an unreasonable silence. The less dramatic the medical explanation sounds, the more easily it can be interpreted as evasive. A physician who focuses on the pregnancy risk rather than debating prehistoric origins may be seen as avoiding the real issue. A blood bank that discusses donation need rather than hidden abilities may appear to be repeating a script. The absence of sensational confirmation becomes, in the conspiracy imagination, confirmation that the subject is controlled. Modern communication has given this process an extraordinary engine. Before the digital age, an individual with Rh negative blood might know little beyond what a doctor or family member explained. Now, within minutes, that person can enter an enormous informal archive of speculation. Videos promise suppressed truths, message boards gather accounts from people comparing dreams, eye colours, body temperature, sensitivity to light, pregnancy histories, unusual feelings, and ancestral hunches. Short-form posts turn a complex blood group system into a dramatic claim designed to survive only a few seconds of attention. Most humans have one factor. Some do not. Scientists cannot explain why. Therefore, something has been hidden. Repetition creates the impression of evidence. If thousands of posts repeat that R-negative people share unusual characteristics, the claim begins to look as though it must have been established somewhere. Even when each post ultimately traces back to other unverified posts, the system rewards the most striking version of the story. A careful explanation of the RHD gene and maternal alloimmunization is important, but rarely feels urgent to someone browsing late at night. A claim that a rare blood type proves descent from an ancient non-human lineage offers a revelation. It invites sharing, debate, indignation, and self-recognition. The more extraordinary the claim, the more attention it attracts, the more attention it attracts. The more people encounter it as though it were a widespread question science has been forced to answer. The evidence has not strengthened. The visibility has. A myth that once might have remained confined to a small occult publication or private group now travels across continents, repeatedly rewritten in new voices, translated into new languages, and presented to millions of people as a forbidden fact discovered just beyond the reach of official history. Certain phrases are especially effective in this environment. They don't want you to know requires no identification of who they are. Scientists cannot explain converts' unresolved details into total ignorance. Doctors will never tell you misrepresents a routine medical subject as concealed. Ancient Bloodline gives a clinical result the atmosphere of inheritance, prestige, and destiny. Negative is treated not as shorthand for the absence of the D antigen, but as a coded sign of something removed, suppressed, or missing from conventional evolution. Once these phrases are placed together, the theory protects itself. If experts reject it, that proves experts are part of the silence. If medical institutions explain Rh incompatibility, they are said to reveal only the controlled public version. If genetics identifies ordinary mechanisms of inheritance, those mechanisms are dismissed as incomplete because they do not answer the mythical question the theory has invented. In reality, medicine has strong reasons to communicate Rh negative status plainly, even if the communication sometimes fails. A pregnant patient needs to know whether she may require anti-D prophylaxis, whether antibodies have already formed, and whether additional care is appropriate after particular pregnancy events. A transfusion recipient needs correctly matched blood. A donor may be encouraged to give because their blood is needed. None of this requires a complete lecture on population genetics or the history of the RH system at every appointment. Medical conversations are shaped by urgency, relevance, available time, and the patient's immediate needs. That can leave people feeling they have been handed only a fragment of a larger story. Sometimes they have. But an incomplete explanation in a rushed clinical setting is not the same as deliberate concealment of human origins. The subject is particularly vulnerable to misunderstanding because the vocabulary can sound more ominous than it is. An Rh-negative mother may be told that her blood is incompatible with the baby's blood, that she could become sensitized, or that her antibodies might attack fetal red cells. Every one of those terms has legitimate clinical meaning, taken outside context. However, they can imply that the pregnancy crosses a profound natural boundary. Incompatible sounds like two kinds of life that should not have joined. Attack sounds as though the maternal body recognizes the child as fundamentally foreign. Sensitized can seem like activation of a hidden condition, yet the same immune principles operate with many red cell antigens and in transfusion reactions among entirely ordinary people. Medical terms describe risk. They do not certify mythology. The idea of a conspiracy becomes even more persuasive when people find genuine failures in healthcare. A woman who did not receive adequate prophylaxis and later becomes sensitized may ask, with reason, why she was not protected. A family may lose a baby in a place where testing or treatment was unavailable, then discover that effective prevention exists elsewhere. A patient seeking rare compatible blood may confront delays, limited inventories, or a system unable to answer urgent questions compassionately. Such experiences can create justified anger, but justified anger does not validate every explanation that attaches itself to it. The real failures are painful enough unequal access, missed care, poor communication, shortages, and the neglect of people whose dangers medicine knows how to reduce. When these failures are absorbed into the claim that Rh-negative origins are being suppressed, attention shifts away from the problems that can actually be corrected. Another source of suspicion comes from the way institutions handle uncertainty. Science is not embarrassed to say that the precise evolutionary reasons for the distribution of Rhd negative status remain under investigation. It can identify the gene, explain the antigen, map broad frequencies, study population history, and still acknowledge that no single model completely reconstructs why the trait persisted at particular rates in every region. To people trained by conspiracy narratives, this admission appears as a crack in the official story. If experts do not know everything, perhaps they know nothing. If they cannot provide an exact ancient origin point, perhaps they are concealing it. But uncertainty is not silence. It is the boundary between what evidence can support and what imagination would like to supply. The refusal to invent a final prehistoric answer is not weakness. It is the discipline that prevents an unanswered detail from becoming a fabricated civilization. A superior caste or an extraterrestrial ancestor? Online communities can nevertheless offer something valuable to people who find them recognition. A person with a rare blood type may enjoy meeting others who share it. A woman affected by alloimmunization may find support among people who understand specialist monitoring and the fear attached to pregnancy after sensitization. A rare donor may gain pride and purpose through contact with others who donate. These forms of connection are not inherently problematic. The danger begins when shared medical experience is transformed into shared biological destiny, and when identity is built around the belief that members possess traits or origins denied to everyone else. A community formed around support can slowly be drawn toward exclusivity. Ordinary coincidences become signs. Doubt becomes evidence of being unawakened. Personal pain becomes proof that outsiders cannot understand. The rare trait stops being one part of life and becomes a total story about who a person is. There is an old human pattern beneath this modern machinery. Across history, groups have claimed descent from special ancestors, divine founders, heroic survivors, sacred kings, or bloodlines set apart. Such claims give identity depth and shape. They say that a person is not merely alive in the present, but part of a meaningful chain stretching backward beyond ordinary memory. Negative mythology does the same thing in scientific dress. Instead of a heraldic emblem or legendary genealogy, it uses a blood test. Instead of a sacred origin preserved by priests or family tradition, it invokes genetics, antigens, suppressed research, and population maps. The technology has changed. The emotional need has not. People continue to seek evidence that their lives are connected to something exceptional, especially when the evidence appears to be carried inside their own bodies. Yet history offers a warning whenever blood is turned into destiny. The belief that inherited biology separates people into purer and lesser groups has rarely remained harmless, even when it begins as personal curiosity or spiritual identity. It can feed ideas of superiority, suspicion toward outsiders, or the fantasy that some unions are dangerous because they cross forbidden biological lines. Rh incompatibility is especially vulnerable to this misuse because it involves pregnancy and risk. An unsupported story can transform a preventable immune reaction into a warning against mixing populations. As though the suffering of infants proves that certain people should remain apart, it proves nothing of the kind. Rhd positive and Rhd negative people belong to one interbreeding human species, and their children are safely born throughout the world. The historic danger lies in maternal sensitization, not in moral or ancestral incompatibility. The conspiracy of silence, then, is less a hidden operation than a cultural machine. It is powered by rarity, incomplete understanding, medical fear, algorithmic repetition, old desires for special ancestry, and real occasions when institutions have failed to explain or protect. It thrives because every correction sounds duller than the claim it must undo. There is no secret bloodline to reveal, only genes, no concealed power, only an antigen difference, no authority forbidding inquiry. Only a body of science complicated enough to be misrepresented in a sentence. Yet the myth is not entirely detached from reality. It feeds upon the genuine gravity of blood. A woman's pregnancy can be affected. A donor's blood can be unusually needed. A family's losses can be linked to a trait passed through generations. A population map can preserve shadows of ancient movement. The myth takes these truths and rearranges them until ordinary human inheritance becomes a message from somewhere beyond the human world. What science offers instead is not a stripping away of meaning, but a different kind of meaning. The body is not less remarkable because it is understandable in parts. A red cell antigen does not need to be alien to connect a modern person with ancestors who survived cold landscapes, migration, isolation, birth, and disease. A preventive injection does not diminish the mystery of pregnancy. It reveals how deeply knowledge can protect life. A rare donor is not special because they possess hidden powers, but because they can choose to turn an inherited accident into another person's survival. The story becomes smaller only if grandeur requires deception. And so the most persistent silence around Rh-negative blood may be the silence people resist most strongly, the possibility that the truth is both extraordinary and entirely human. No guardians have hidden the origin of a superior lineage. No ancient record waits to prove that the minus sign marks visitors from the stars. What has been hidden, for most of history, is hidden only because it is microscopic a surface antigen, an antibody, a gene passed without awareness from parent to child. Once brought into light, it tells a story not of separation, but of how fragile and interconnected human lives have always been. The myth asks people to distrust the ordinary explanation. The ordinary explanation, followed far enough, leads back through medicine, grief, ancestry, and deep time to a truth no conspiracy can improve. Human blood is varied. Human history is unfinished, and even a small inherited difference can become large enough to shape the stories people tell about who they are. The collapse of a myth does not end the investigation. Once claims of alien inheritance, hidden powers, and deliberate suppression are placed where they belong, outside credible evidence, the Rh-negative story does not become closed or uninteresting. It becomes available for better questions. A gene can be identified without every stage of its history being known. A blood group distribution can be measured without revealing exactly which ancient event shaped each rise and fall in frequency. Medicine can understand how an antibody attacks fetal red cells and still leave evolutionary puzzles unresolved. Science does not require a mystery to disappear before it can speak clearly about what is known. It requires only that the border between evidence and speculation remain visible. One of the basic questions is deceptively simple. When did the genetic patterns associated with common Rhd-negative blood arise? And how did they spread in many people of European ancestry? Rhd-negative status often results from the absence of the Rhd gene. That genetic arrangement must have emerged somewhere along the long path of human reproduction, through the kinds of rearrangements and deletions that occur in genomes over generations. But finding a trait in modern populations does not instantly reveal the date or setting of its earliest appearance. The first carrier left no written account and almost certainly had no idea anything about their blood was unusual. They lived, reproduced, or did not reproduce, moved through a landscape, and passed a small inherited feature into a future they could not imagine. By the time the trait becomes measurable, thousands of years of migration, mixture, population expansion, and loss may have rearranged its distribution beyond any simple reconstruction. Ancient DNA has changed the study of human population history with astonishing speed. Genetic material recovered from old bones and teeth has revealed that the human past was far more mobile and entangled than many earlier theories assumed. Hunter-gatherers, early farming populations, steppe-associated pastoralists, and later historical communities did not simply replace one another in clean lines. They met, mixed, separated, expanded, and left descendants in proportions that varied across regions. In principle, ancient genetic data can also help trace blood group variants through time. But the task is not always straightforward. Ancient DNA is often fragmented and damaged. Not every burial preserves readable material. Not every region or time period is equally sampled. A few ancient individuals cannot stand in for an entire population. And a population living in one place at one date cannot automatically be treated as the direct source of a modern frequency pattern. What survives in the record suggests roots of continuity and change, but the map remains filled with gaps. The Rh genetic region presents its own technical difficulties, because RhD and RhCE are closely related genes, and because the region can contain deletions, hybrid arrangements, and variant forms. Determining the precise Rh phenotype from ancient genetic material may demand unusually careful analysis. Modern clinical testing can examine living blood cells directly, observing whether particular antigens appear on their surfaces. Ancient remains offer no functioning red cells to type in a hospital laboratory. Researchers must infer genetic possibilities from preserved DNA when sufficient relevant sequence exists. and distinguish true absence or variant structure from the ordinary damage and incompleteness of ancient samples. A badly preserved genome cannot be forced to answer a question it does not securely contain. This is one reason the origins of aurora at negative frequency do not reduce to one dramatic skeleton, or one lost tribe suddenly identified by science. Even with better ancient sampling, the evolutionary puzzle contains an apparent tension. The RHD negative phenotype can create a reproductive disadvantage. Under certain circumstances, an RHD negative woman may become sensitized by an RHD positive fetus, endangering future RHD positive pregnancies. Before prophylaxis and modern care, severe cases could end in fetal or newborn death. If natural selection tends to reduce traits that lower reproductive success, why did RHD negative blood persist? And why did it become relatively common in parts of Europe? The question is reasonable. It has sometimes been presented as though biology cannot answer it without invoking an extraordinary origin. But evolution often preserves traits with disadvantages when those disadvantages apply only in particular circumstances, appear late in reproductive sequences, vary among individuals, or are balanced by population history and chance. The reproductive cost of RHD negative status is not automatic. The mother must be RHD negative. The fetus must carry the relevant D antigen. Fetal red cells must enter her circulation in a way that provokes sensitization, and a later D-positive fetus must then be exposed to clinically significant maternal antibodies. A woman partnered with an RHD-negative man would not face anti-D disease in an RHD-negative fetus. A woman with an RHD-positive partner might have children who do not all inherit D if the father carries one D-associated and one D-negative genetic form. Some women are not sensitized despite exposure. Some affected infants historically may have survived. The damage, though terrible in individual families, is spread unevenly across carriers and across generations. Natural selection acting against such a trait may therefore be weaker than intuition suggests, particularly when other forces push its frequency upward in a small or isolated population. Genetic drift is one such force, but it is difficult for people to accept because it denies that every distinctive trait must have a purpose. The human mind prefers a reason, perhaps, or a negative blood protects against an ancient infection. Perhaps it improves survival in a harsh climate. Perhaps it reflects an important biological difference maintained for advantage. These possibilities can be studied where evidence suggests them, but they should not be assumed merely because a trait exists. In small populations, frequencies can shift dramatically through chance. A group founding a settlement may happen to contain more RHD negative carriers than the wider population. A later expansion can spread that elevated frequency across a region. A famine, epidemic, or conflict can remove some family lines and leave others disproportionately represented. Over enough generations, a trait with a limited cost can become common without ever having granted an advantage. Researchers have explored whether Rh status may influence susceptibility to infections or other aspects of health, because blood cell surface proteins and immune interactions can sometimes carry consequences beyond the best-known clinical setting. The history of human genetics includes clear examples of such relationships. Certain red cell variants influence malaria risk. Some blood group distributions may reflect past disease environments. It is therefore not unreasonable to ask whether the Rh system has experienced selective pressures not yet completely understood. But asking is not answering. Associations detected in individual studies must be replicated, separated from ancestry and environmental confounding, and shown to matter strongly enough to explain population frequencies. To date, no single demonstrated survival benefit offers a settled explanation for the high prevalence of common RHD negative forms in particular European populations. Scholars still debate what combination of drift, population history, and possible selection shaped the pattern. This is a very different uncertainty from the one promoted in fringe accounts. Science is not standing helpless before a blood type whose nature it cannot identify. It can describe the Rh proteins, the genes involved, the inheritance patterns, the antibodies, the pregnancy risks, the transfusion consequences, and the broad geography of frequencies. Its open questions occupy a more demanding level exactly when variants arose, how they moved through prehistoric and historic populations, whether some effects beyond pregnancy altered their success, and how different genetic backgrounds produce similar clinical results in different regions. These are questions of resolution, not of basic reality. The map is incomplete because deep human history is difficult to recover, not because the true answer has been removed from view. There are also questions that matter immediately rather than archaeologically. The ARCH system is still medically complex. Routine positive or negative typing captures the most important distinction for many patients, but it does not reveal every variant relevant to transfusion and pregnancy. Molecular testing can help identify weak or partial D forms, guide whether a patient should receive RHD negative blood, and clarify whether prophylaxis is needed in certain situations, in diverse populations. Better understanding of RHD variants can prevent both unnecessary treatment and avoidable sensitization. Blood banks continue refining donor typing because patients with antibodies may require matches extending well beyond ABO and D. For those patients, scientific progress is not an abstract debate over origins. It is the difference between a compatible unit being found quickly and a dangerous delay. Pregnancy medicine also continues to evolve. In some settings, testing can identify fetal RHD status through fragments of fetal DNA circulating in the mother's blood, allowing care to be tailored more precisely when available and appropriate. An RHD negative mother carrying an RHD negative fetus has no risk of anti-desensitization from that fetus, while one carrying an RHD positive fetus may benefit from prophylaxis according to clinical guidance. Such approaches reveal the trajectory of real medical inquiry away from broad assumptions and toward increasingly exact knowledge of each pregnancy. The history begins with families losing babies for reasons no one could see. It moves through blood group discovery, antibody testing, preventive injections, fetal monitoring, and now genetic information drawn from a maternal blood sample. Nothing about that path suggests a science intent on concealment. It suggests a science steadily learning to see what once remained invisible. The question of origin remains attractive because it promises to place every Rh negative individual inside one ancient narrative. But a trait need not come from a single culturally meaningful people in order to have history. The gene does not carry a label saying Basque, Ice Age survivor, royal line, or lost civilization. It may have risen in frequency within communities that later disappeared into larger populations. It may have crossed boundaries through marriages never recorded. It may have become common where chance favoured its carriers and declined where their lines faded. Ancient DNA may clarify portions of that route, and future research may alter current interpretations. That is the proper humility of historical genetics. Every living frequency is an outcome, but the exact chain of events producing it is not always fully recoverable. There is a temptation, especially in mystery storytelling, to treat an unresolved detail as a locked door behind which the most dramatic possible answer must be hiding. For negative blood teaches a different lesson. Sometimes an unresolved detail is simply the edge of evidence. Beyond it lie not secret masters or impossible ancestors, but lost births, unrecorded migrations, destroyed burial grounds, populations sampled only in fragments. A molecular process is too small to leave an obvious trace in the archaeological world. The past is silent not because it conspires, but because most human lives passed without documents, without preserved remains, and without knowing which of their inherited traits would fascinate distant descendants. If the deeper origin of RHD negative frequency is one day reconstructed more clearly, it will likely come not as a revelation that overturns humanity, but as a more detailed human story. Perhaps researchers will identify when a common deletion was already present in ancient Western Eurasian groups. Perhaps they will map how it moved with farming communities, survived local bottlenecks, or increased in particular regions after later isolation. Perhaps previously suspected selective effects will prove meaningful, or perhaps drift and demographic history will remain the stronger explanation. Each discovery would add texture to the narrative, not transform the trait into evidence of a hidden species. Good science does not fear the result because it does not require the answer to flatter a theory. And there is a final irony in the demand for Rh negative blood to conceal some extraordinary power. Its most extraordinary power has already been demonstrated, though not within the blood itself. It is the power of inquiry to move from confusion to prevention. People once watched newborns sicken and had no explanation. Researchers then recognized that blood carries inherited markers, that antibodies can cross the placenta, that the D antigen is central to many cases, and that sensitization can be prevented. Blood banks learn to match donors with increasing precision. Obstetric teams learn to protect pregnancies once thought doomed. Rare donor networks learn to link strangers across the world. These advances are less dramatic than a claim of ancient gods or visitors from the stars, but they have done something myths cannot do. They have allowed children to live. The remaining mystery is not a vacuum waiting for fantasy. It is a field of honest questions, each tied to evidence, each limited by what time has preserved, each still capable of revealing something about the human past. Or a negative blood has ancient origins because all inherited human variation does. It carries a history of migrations and chance because every population does. It once exacted a severe cost in families because biology can be indifferent to love. And it now survives in an age capable of understanding much of its danger, while continuing to investigate the distant path by which the minus sign reached the present. Science has not silenced that mystery. It has made it possible to ask the mystery without mistaking it for a myth. After every layer has been stripped away, the frightened speculation, the alien ancestry claims, the promises of hidden ability, the language of forbidden blood and secret authorities, the Rh negative mystery remains. It is simply not the mystery the legends promised. No hidden archive is needed to explain why it matters. No suppressed dynasty waits beneath the ruins of an ancient civilization. No unusual sense, intelligence, or spiritual faculty appears in the blood because a person lacks the D antigen. What remains is quieter, and, in the end, far more consequential an inherited variation that travelled through human populations for untold generations. Once placed mothers and infants in mortal danger without explanation, and eventually forced medicine to understand blood not as a symbol of identity alone, but as a precise biological language written on the surface of living cells. The minus sign survives because human variation survives. Every person alive carries countless inherited details shaped by ancestry, chance, migration, isolation, disease, and the simple fact that some family lines continue while others disappear. Most of those details never become stories. They influence an enzyme, a receptor, a pigment, a response to food, or a tiny molecular feature no one notices. RHD negative status became different because the consequences could be seen even when the cause could not. A newborn turning dangerously yellow, a fetus swelling before birth, a mother whose first child lived while later infants died. A transfusion meant to rescue a patient instead producing fever, collapse, and destruction of blood cells. Long before anyone could identify the antigen, the trait wrote itself into human experience through fear and loss. Once science gave the pattern a name, It also gave later generations a way to imagine meanings beyond the evidence. Perhaps that is why the legend has lasted, where negative blood feels as though it ought to mean something enormous because, under certain circumstances, it has done enormous things. It has decided whether a pregnancy required protection. It has determined which donor unit could safely enter a bleeding patient. It has connected families with a history of unexplained loss to an answer arriving too late for those already buried. It has made strangers valuable to one another through rare donor systems and emergency transfusion. Yet none of those realities require supernatural origin. Meaning does not vanish merely because the mechanism is natural. A storm is no less overwhelming because its winds can be measured. A glacier is no less ancient because its movement can be calculated. A red cell antigen is no less important because it arises from genes rather than from myth. The ancient origin question also settles into a more honest shape. Or a negative blood is ancient, not because it proves descent from one vanished elite, but because every inherited trait in living humans has a journey behind it. Somewhere in deep time, genetic variations affecting expression of the D antigen emerged and entered human family lines. Those lines moved through changing landscapes, through cold periods and warming valleys, through farming expansions, local isolations, marriages, wars, epidemics, voyages, and migrations too small ever to appear in chronicles. In some populations, particularly in parts of Europe, common RhD negative forms became relatively frequent. In others, they remained uncommon or appeared through different molecular variants within the complex Rh system. The pattern is a real clue to population history, but it is not a single stamped seal of origin. It is more like a faded thread crossing a vast woven fabric visible in places, interrupted in others. Valuable when examined carefully, ruined when pulled free and claimed to be the whole design. The Basque Association, so often placed at the centre of the legend, illustrates the difference between mystery and invention. Their language is genuinely extraordinary in the modern European landscape. Their population history does show meaningful distinctiveness and continuity. The relatively high Rh negative frequencies are a real subject of historical interest, but those truths do not isolate them from the rest of humanity. Nor do they turn Rh negative people elsewhere into members of a secret biological nation. The Basque story is not powerful because it offers proof of an unearthly bloodline. It is powerful because it shows how culture and ancestry can endure without remaining untouched, how a language can survive empires and borders, how a population can preserve distinctive frequencies while still participating in the movements, mixtures, and transformations of human history. Their blood does not point outside the human past, it points more deeply into it. The claim of hidden genetic power dissolves even more completely under scrutiny. There are no credible grounds for linking ordinary RhD negative status with psychic perception, superior intelligence, unusual emotional sensitivity, or a body fundamentally unlike that of RhD positive people. There are only human beings whose red blood cells either display or do not display a particular antigen in its ordinary form, along with the many variants that make actual transfusion medicine more complicated than a plus or minus sign. If there is power here, it belongs not to a select group, but to knowledge. The knowledge that identifies an antibody in a pregnant patient, the knowledge that protects an unsensitized mother with anti-D prophylaxis, the knowledge that monitors a fetus for anemia and delivers blood before birth when necessary, the knowledge that locates a rare compatible donor before a patient's time runs out. These achievements do not flatter anyone's ancestry. They do something greater, they prevent suffering. The conspiracy of silence changes shape as well. There was, for most of human existence, a silence around Rh disease, but it was the silence of ignorance. Mothers lost children without knowing why, because the tools needed to see the cause did not exist. Later, there were gaps of access and failures of communication, and such failures still matter wherever preventable disease continues because patients cannot obtain screening, prophylaxis, specialised care, or safe blood supplies. Those are not imaginary concerns. They deserve attention precisely because they are real. But the scientific account of Rh negative blood is not concealed. It is found in the history of blood group discovery, in obstetric practice, in genetics, in haematology, and in the protocols that protect pregnancies and transfusion recipients every day. The truth has not been hidden because it lacks the theatrical satisfaction of the myth. It has simply been spoken in the careful language of medicine rather than the dramatic language of revelation. And still, one can understand why people continue to search for more. A blood type is not like a distant historical date or a ruined city glimpsed behind museum glass. It is within the body. It belongs to parents and children. It may emerge suddenly in a crisis, carrying instructions about pregnancy, donation, or danger. To discover that one's own blood is relatively uncommon can feel like receiving a fragment of private history, a message inherited from people whose names have vanished. In one sense, that feeling is not wrong. The trait did come through ancestors. It did survive the same centuries they survived. It does connect the present body to the long chain of birth that produced it. The mistake lies only in believing that connection must lead to something beyond humanity in order to be profound. What survives in the record suggests a different kind of hidden truth. Human life has always depended on differences people could not see. A mother in prehistory might have carried the same RhD negative pattern as a mother entering a clinic today. One lived before any person could warn her that a later child might be endangered. The other may receive a blood test and a preventive injection that allows danger to pass without consequence. Between them lies no change in the fundamental blood trait. only the slow accumulation of understanding. The antigen is ancient. The antibody response is ancient. The vulnerability is ancient. What is new is the ability to recognize it in time. That ability should not be taken lightly. Modern medicine is often experienced through fluorescent rooms, forms, needles, waiting, and routine instructions. Its victories can look mundane from inside the systems that deliver them. But consider what anti-deprophylaxis truly represents. A species that spent almost all of its history unable to explain why certain babies died has learned to identify one of the causes before the harm begins and to interrupt it with a treatment given to the mother. A tragedy that once repeated silently in families can now, in many cases, be prevented so completely that the protected child never knows a danger was waiting. This is not a lesser story than one about ancient powers. It is a story of human beings finally becoming able to defend themselves against a sorrow carried invisibly through their own inheritance. Our negative blood therefore belongs to neither fear nor worship. It is not a stain and not a crown. It is not evidence that someone is less human, more human, older in spirit, or biologically separate from the people around them. It is a normal variation with important consequences in specific medical circumstances, a trait whose distribution raises legitimate questions about population history, and a reminder that the body can carry ancient information without turning that information into destiny. An Rh-negative mother can carry an Rh-positive child safely. An Rh-negative donor can save the life of an unknown recipient. An Rh-positive person can share ancestry, love, family, and future with an Rh-negative person without crossing any forbidden boundary. The old danger was real, but the division imagined around it is not. At the end of the search, the rare bloodline does not lead upward toward gods or outward toward visitors from elsewhere. It leads backward through ordinary families, through mothers and fathers, births and losses, journeys and settlements, bodies surviving long enough to pass their variations onward. It leads into the laboratory, where a reaction in a small sample of blood exposes a danger that had operated unseen for millennia. It leads into the maternity ward, where an injection prevents immune memory from becoming grief. It leads into the blood bank, where carefully labelled units wait in cold storage because another person may need precisely what one donor carries. And it leads, finally, into a simpler recognition the human past is strange enough without invention. Our blood is full of traces, not of secret superiority, but of all the fragile ways life has continued. The mystery remains because no gene can tell the complete story of every journey that brought it into the present. Scholars may continue to refine when and where particular Rh variants spread, how drift and selection interacted, and what ancient populations contributed most strongly to modern frequencies. New samples may revise old assumptions, better genetic tools may recover signals now out of reach, but the central truth will not require a cover-up to explain it. Our negative blood is human blood rare in some places, more frequent in others, medically significant, historically consequential, and entirely part of the immense inheritance shared by a species whose greatest secrets have so often been hidden not by conspirators, but by the limits of what it could once see. For all the noise that gathers around rare blood, the story ends quietly. Somewhere. A child sleeps after being born into a world better prepared than the one that received countless children before. Somewhere. A mother leaves a clinic with a small mark on her arm and no visible proof of what has been prevented. Somewhere. A donor sits beneath ordinary lights while blood moves slowly into a collection bag, unaware of the face or name of the person it may one day sustain. None of these moments looks like legend. There are no vanished temples, no sealed vaults, no revelation written across the sky. There is only life continuing because a hidden difference has finally been understood. The ancestors who carried that difference knew nothing of antigens or genes. They walked through rain and winter, tended fires, crossed valleys, buried their dead, held their infants, and passed forward a trait as silently as they passed forward the shape of a hand or the memory of a face. Some mothers among them must have endured losses whose explanation lay impossibly far in the future. Their grief leaves almost no trace that can be named with certainty now. Yet the knowledge gained centuries later reaches backward in a small act of justice. It tells us that many of those deaths were not punishment, not weakness, not proof of a cursed line. They were biology unseen. Time does not return what it has taken. It allows only understanding, and sometimes prevention. That is enough to change the meaning of an inheritance. The minus sign no longer needs to be feared as a secret verdict or celebrated as a mark of hidden power. It can be carried simply as one ancient variation among countless others, a reminder that human bodies preserve histories too small for monuments and too persistent to vanish. And beyond the hospital window, beyond the laboratory glass, the old landscapes remain mountains gathering rain, coastlines receiving the tide, paths crossing valleys where generations moved without knowing what they carried. Their blood has reached the present, not pure, not supernatural, but living. In that unbroken movement lies the only hidden power the story ever needed: fragile life, passing through darkness, learning at last how to protect what comes next.